Jung.
Yeah, everybody's muted for now. Yeah, just, it's good to see you. We'll start in a minute.
Yes, and everybody should be having the camera off during the meeting, otherwise the teams will get very slow.
Good afternoon to all.
I guess it's soon time to get started. The time says it's 2 o'clock in the afternoon here in Oslo. And with that, I would like to really welcome you all to the webinar, Healthy Nursery Plants, the Key to Healthy Horticulture, Forestry, and Nature Restoration. I have to say that we had a truly overwhelming interest in people registering for this webinar, with over 800 people registering from 63 different countries across 400 different affiliations. And this is really delightful. I think it really shows how important it is to keep nurseries healthy and what implications it has if it doesn't. And it shows that this is an issue not just related to nurseries themselves, but that is an issue related to the whole value chain, from policymakers to plant health inspectors, authorities, governmental institutions, and universities. So welcome all. Thank you very much for joining us. To make sure that the content of this webinar is available also to those who would like to join but are unable to because of the time differences, we are recording this webinar and it will be posted on the PurPest website. We will send the link to all the people who have registered. Some people might need a sort of certificate of attendance. We can organize that. If you do need this certificate, please send us an email.
Yeah, that's fine.
In order to keep this webinar on track and really avoid any noise in the background, I ask you to keep your camera and your microphone muted. Turn it off, because otherwise, with these many people, the system might break. There are no questions scheduled right after each presentation, but please post all your questions and comments in the chat. Please write in English when you do that, and give your name, your affiliation, and the speaker you want to address the question to. This really helps us from the organizing committee to select those questions that are relevant for most of us, and that we can then discuss in the discussion section at the end of the webinar. The chat is only to be used for questions and comments related to the webinar, so please abstain from any inappropriate comments unrelated to the topic. So far for the formalities. As coordinator of a new project, which is called PurPest, Plant Pest Prevention through Technology-Guided Monitoring, I really am very interested in moderating this webinar, because I truly believe that it is always better to look, even if you might not like what you see, than to simply look away and let catastrophe or calamities take its course. So I'm really looking forward to hearing from Dr. Thomas Jung, talking from Mendel University, who will show the devastating effect of Phytophthora on nurseries, landscapes, and forests, before we hear from Dr. Bruno Scanu from the University of Sassari about the best management practices, followed by Dr. Ana Pérez-Sierra from the Instituto Valenciano de Investigaciones Agrarias, who will lead us towards healthier nurseries. We have a short coffee break then at about 3 o'clock, and then we continue with Dr. Sarah Green from Forest Research at the Northern Research Station of the UK, who will discuss how to identify the risk of Phytophthora infection in British nurseries. Dr. Tyler Bourret from PhytoWorld and Dr. Susan Frankel from the USDA Forest Service will share their experience on how to prevent Phytophthora species spreading to native and horticultural plants in California. Before Dr. Malcolm Catlin, director from the Plant Healthy Scheme, will describe how valuable it is really to have a voluntary nursery certification scheme and how it works. After that, we have time for the discussions. So I think with no further delay, just again, keep your microphones muted. Everybody mute your microphone except for the speaker. Dr. Thomas Jung, I would like you to unmute your phone, not your phone, but your microphone, start your camera and share your presentation for our first part of the webinar.
Okay. Thank you, Andrea, for the introduction. I will start the screen sharing. So, okay. You see my screen? Yes. Okay. Ladies and gentlemen, dear colleagues, thank you very much for participating in this important webinar. The nursery infestations in Europe are a huge problem, and also in other countries, and they spread lots of Phytophthora species to very sensitive natural environments. This webinar here is to bring people together to find solutions for this problem and not to blame anyone for it. So I hope we can initiate.
Thomas, you're muted. Can you please unmute yourself?
Can you hear me now?
Yes. Yeah.
I'm very sorry for that. So then I start the screen.
Your screen was fine. It was just suddenly when you said we're trying to work together on this.
Okay. So thank you very much for everyone participating in this important webinar. The nursery infestations in Europe and elsewhere are a huge problem, spreading lots of harmful Phytophthora species into sensitive environments. This meeting here is to bring people together to start a process to solve this problem. It is not for blaming anyone about it. I talk now about the epidemic Phytophthora diseases of forests and natural ecosystems and the importance of the nursery pathway in the spreading of the disease. I show you this very simplified phylogenetic tree to just show you that Phytophthora species that belong to the Oomycetes are in an own kingdom, the Chromista, and they are not related to fungi. They are more related actually to brown algae and diatomea. Phytophthora species produce very typical irregular coralloid hyphae. All Phytophthora species produce thick-walled oospores inside of oogonia. These are resting structures that can survive for many years in the soil without any host. Approximately 30% of the Phytophthora species also produce chlamydospores, vegetative survival structures that can also survive for a few years in soil. The main infective propagule of Phytophthora species are zoospores that are released from sporangia. And in airborne aerial Phytophthora species, the sporangia that form on aerial parts of plants are spread like conidia of fungi through the air by wind and by rain splash. Here you see a sporangium of Phytophthora plurivora, one of the major pathogens here in Europe, and it is releasing zoospores. Zoospores are biflagellate and they move actively through the soil water and river water. They are chemotactically attracted to fine roots, following a concentration gradient of organic acids released by the fine roots and accumulating in the elongation zone of the fine roots. And you can see here hundreds of small zoospores that have encised on the surface of this beech root, infecting and killing the beech root. Zoospores can move approximately up to 1 week in the soil. Why shall we care about Phytophthora pathogens? Soil-borne Phytophthora species are responsible for more than 70% of all known fine root diseases and more than 90% of all bleeding bark canker diseases of woody plants. They are causing many of the most devastating epidemic diseases of horticultural crops, forests, and natural ecosystems worldwide. And many Phytophthora pathogens can infect between 100 and up to 5,000 plant species. I give you the take-home messages in the beginning. Due to the lack of coevolution, native tree, shrub, and crop species are not adapted to invasive introduced Phytophthora pathogens. Almost all nurseries are infested with multiple invasive and aggressive Phytophthora pathogens, and therefore almost every planting activity is spreading aggressive Phytophthora pathogens to the wider environment. We can only solve this with a science-based nursery accreditation scheme to prevent that the large-scale nature restorations cause a phytosanitary disaster. In this graphic, you can see that between 1876, when Phytophthora infestans was first described, and the mid-1990s, the number of described Phytophthora species was slowly and steadily increasing up to 50 species in the mid-1990s. And then from 1995 on until now, in 30 years, the number was exponentially exploding to now 265 Phytophthora species. The reason for this unprecedented rise in species numbers are studies of natural ecosystems, service in unsurveyed regions in Asia, Africa, and the Americas, and increasing introductions of Phytophthora species by global boundary. Every one of you knows that Phytophthora species can cause epidemic diseases in clonal horticultural crops. This is an example of late blight of potato and tomato caused by Phytophthora infestans. Another example here is patch mortality in a sunflower field caused by Phytophthora rosacearum after heavy rain and water ponding in this field. And you see here the root rot and the collar rot of the sunflower plants. But Phytophthora species also cause devastating epidemics in forests and natural ecosystems. And in parallel to the increase of the number of described Phytophthora species, also the number of known epidemics of forest diseases was increasing slowly until the mid-90s, and then exponentially increasing up to a number of 41 known Phytophthora epidemics in forests. The most well-known species, and probably also the best studied disease, is the dieback of diverse eucalypt forests in Australia. It is caused mainly by Phytophthora cinnamomi, Phytophthora multiflora, and then also a range of other Phytophthora species. The Phytophthora and the disease, once introduced to an ecosystem, move in a disease front of 50 to 100 meters advancement every year through the forest. They kill the understory, and then years later also the canopy of the big trees. That's why Phytophthora cinnamomi and dieback of forests is called the biological bulldozer in Australia. An airborne aerial Phytophthora species is Phytophthora ramorum, and as everyone of you knows, it's causing sudden oak death in California. The pathogen is producing sporangia on the leaves of understory shrubs, and from there the sporangia are spreading in high numbers onto the stems and branches of oaks and 10 oak trees, causing multiple cankers of the stems and branches, leading to wilting and the collapse of the oak and 10 oak stems. The same pathogen is causing a similar disease with epidemic extent in the UK called sudden large death. The only difference to sudden oak death is that the pathogen is sporulating on the large needles, and from the large needles then spreading with sporangia onto the stems and the branches of large and killing the trees very quickly. Also, a well-studied disease is now European oak decline or European oak declines. Since the 1990s, many studies have been performed. You see here Quercus robur, a mature tree with dieback symptoms and chlorosis of leaves. It is caused by extensive losses of lateral roots and fine roots caused by Phytophthora plurivora and Phytophthora quercina in the range of up to 20 different Phytophthora species. Besides the fine root losses, the pathogens also cause cankers, as you can see here and here. In interaction with the severe droughts in Southern Europe, cork oak and holm oak decline can be very devastating, leading to massive death in patches and stand levels, and leading also to deforestation of landscapes, as you can see here in Portugal. Besides the root damages, Phytophthora are causing in cork oak also bleeding cankers on the lower stem. This is also a collapsed cork oak stand in Sardinia. The trees died because Phytophthora cinnamomi was girdling the collar of these trees. All across Europe, we were studying 121 nursery stands and 77 nurseries. In this study that was published in 2016, and the infestation rates were 87 to 89%. And in the young plantations, younger than 20 years, the infestation rate was approximately the same. The nurseries and the plantations had the same spectrum of Phytophthora species, and most prominent were Phytophthora canvivora, cinnamomi, plurivora, and quercina. These are also the main drivers of the decline in the field. You cannot hear me? People are complaining on the chat that they
We can hear you fine, Thomas. I think
I can hear you.
Okay, because
That's fine.
people were chatting that they can't hear me.
Yeah.
Okay, now I continue. Here you see in the nurseries typical mortality of Quercus ilex plantlets. They are in containers standing on the ground and were probably infested with rain splash of and irrigation splash of zoospores onto the containers. And this is an example of cocoa plants with chlorosis and mortality in Portugal, also caused by Phytophthora cinnamomi. In Eastern Europe and Central Europe and parts of Southern Europe, most forest nursery plants are still produced in fields in the ground, which is a very devastating way of producing nursery plants, because every Phytophthora that gets introduced in such a field will forever stay there, and over the years more and more different species of Phytophthora are introduced into these fields. And you can see here these Quercus petrea plants wilting and dying because of Phytophthora plurivora and Quercina. This is a typical afforestation of cork oak in Portugal, and you see the collapse of the trees because of Phytophthora cinnamomi collar rot infection. And this is an afforestation with Quercus robo in Germany. You see the chlorosis and the dieback of the plants caused by root rot by Phytophthora plurivora. Very devastating and very concerning is European beech decline all across Europe. It's caused by interactions between invasive Phytophthora species and climatic extremes. That means severe rains and floodings interacting with droughts. This is the dieback of an alpine forest in Bavaria, and it is caused by root rot and aerial bleeding cankers caused by Cambivora and Plurivora. This is a lowland beech forest in Austria in the Vienna region, and it's collapsing dramatically because of Phytophthora Cambivora collar rot infections. In beech, as already mentioned, beside the extensive losses of fine roots and lateral roots, also collar rot infections occur with these bleeding cankers and the advancing front here in the bark, and aerial bark cankers, bleeding cankers that can reach up to 20-25 meters in the canopy. Here we also tested the nurseries across Europe, and the infestation rates in the nurseries were approximately 80%, and in young plantations even 93%. Most widespread were Phytophthora cactorum, Cambivora, and Plurivora, and these are also the most aggressive species to the old growth stands of beech and in pathogenicity trials. You see here a forest nursery field in northern Germany with extensive mortality of the seedlings after heavy rain and ponding of water on the soil. This is an afforestation in Germany, and you see the decline and the chlorosis of the leaves in this beech tree. This beech tree also had collar rot symptoms caused by Phytophthora cambivora. This is a very expensive and very old amenity tree planted in Slovakia in a garden. You see the chlorosis, you see the thinning of the crown, and this tree also had collar rot caused by Phytophthora plurivora. The longest researched and longest known forest disease caused by a Phytophthora species is ink disease of chestnut Castanea sativa, and the main causal agents are Phytophthora cinnamomi and Cambivora. The pathogens cause root rot and collar rot, and it is staining the roots and the surrounding soil in black color. That's why the disease is called ink disease. Here you see the advancing inner bark lesion. Here the infestation rate in the nurseries was even above 90%, and in the plantations 72%, and it was mainly Phytophthora cambivora and cinnamomi. Phytophthora cinnamomi shockingly was found in more than 50% of the nursery fields and plantations of chestnut. The epidemic Phytophthora mortality of alnus forests across Europe is a textbook example for large-scale failure of well-intended ecological restorations. The riparian alder stands show dramatic mortality rates all across Europe, with mortality levels reaching 70 and up to 90%. It's a root and collar rot disease caused by the hybrid Phytophthora X alni. The disease and the mortality rates are particularly severe in swamps with standing water all year round, as you can see here. The disease is widespread across Europe, except of Southern Europe, Italy, and the Balkan and Greek area. Here you see the rusty typical exudations of the collar rot lesions on a mature stem caused by Phytophthora alni. In 2002-2003, we performed a statewide survey in Bavaria along 20,000 kilometers of rivers and streams, and found the disease distributed, as you can see, with the red stretches of rivers along most of the rivers already in 2003. We then traced back the source of the inoculum, and we made a forest survey in the forest alno stands in Bavaria. We surveyed 3,300 forests, and of these 1,100 alnus forests were infected, and 92% of the infected forests were planted and younger than 20 years, an obvious indication that the disease was coming from the nursery plants. This is a typical afforestation, a first afforestation with Alnus glutinosa, and you see here this chlorotic tree and a dead tree, and these trees had the typical rusty exudations of the color rot lesion and the flame-shaped appearance of the inner bark lesion. This is Professor Jörg Schumacher from Germany, also a plant pathologist, and together we did a survey in nurseries in Germany. These are healthy-looking 2-year-old Alnus glutinosa plants. We surveyed them randomly, put them into water buckets to stimulate the Phytophthora to spread and cause disease. And after 1 to 2 weeks, we found in many of the plants color rot lesions and root lesions, and we could isolate from them a lot of Phytophthora species in 77% of the nursery fields and the plantations, and very widespread was Phytophthora alni, the causal agent of the epidemic, but also other harmful Phytophthora species. In Patagonia, in South America, you can see here a dramatic landscape level mortality of Austrocedrus chilensis, a native conifer there. It's caused by the introduced pathogen Phytophthora austrocedri, and it is a root and color rot disease again. And you see here the advancing front of the lesion in the inner bark. The same pathogen was introduced years later into restoration plantings of Juniperus communis woodlands in northern Britain. And you see here the same symptoms caused again in the junipers, resinous cankers and inner bark lesions, and a dramatic mortality of the old juniper plants. This pathogen, Phytophthora austrocedri, is very widespread in the ornamental nursery trade in Europe now. These are examples from plants sampled in garden centers all over Croatia, and you see the typical wilting symptoms and necrosis. All these plants were imported to Croatia with valid EU plant passports, showing that the plant passport system, based on visual inspections, is not working for soil-borne pathogens. The same symptoms you can see in Camisiparis plants, also imported to garden centers in Croatia with plant passports. Here on the pots, you see the wilting symptoms, and this is Phytophthora lateralis. Phytophthora lateralis is causing dramatic wilting and dieback of plants and chlorosis in hedgerows in the UK, and it is causing a root and color rot. And you see the advancing front of the rot in the inner bark. Here is a typical example of a hedgerow planted in Croatia, also killed by Phytophthora lateralis. Also, pine species are highly susceptible to Phytophthora species, especially Phytophthora cinnamomi and Pseudocryptogaea. This is an example from an afforestation in Sardinia with small-sized needles and chlorotic needles because of root losses caused by Phytophthora cinnamomi. Phytophthora cinnamomi also has killed these plants here in the nursery in Spain. And again, the containers were standing on the ground and probably splash infested with Phytophthora cinnamomi spores onto the containers. This is a nursery in Portugal, and you can see Pinus radiata seedling showing chlorosis and mortality. The reason for this infection by Phytophthora cinnamomi was most likely that the containers are too close to the ground, only 20 centimeters, and rain splash of inoculum onto the containers can occur. In addition, the nursery was using unfiltered river water for watering the plants, and river water is full of Phytophthora species. All over Europe in the last 25 years, 2,500 beds and container stands in 850 nurseries of 20 countries were tested in multiple studies, and these are only a few papers by our group, and we will hear later from other groups similar results. The Phytophthora infestation rates were 80 to 100%, and this was with classical isolation methods. With metabarcoding, you find even more Phytophthorus. 124 Phytophthora species were confirmed, of which 109 are considered as invasive aliens. And this is showing you the result from 2,525 planting sites across Europe of, in total, 470 different woody species. And the infestation rate was 66%. Most widespread Phytophthora plurivora, cinnamomi, nicotianae, Cryptogaea, Cactorum, alni, Sidrophthora, Cambivora, ramorum. This is a recently published study from Czech Republic where we use metabarcoding for a nursery survey, and we show in this diagram the comparison between an ornamental nursery with imported plants and a forest nursery with local production of plants. And in the ornamental nursery with imported plants, 24 known and 8 new unknown Phytophthora species with unknown aggressiveness and host ranges were found. And in the forest nursery, only 2 Phytophthora species that were known, but they were also aggressive pathogens. In this ornamental nursery, we also tested the irrigation system. And in the river water, before it was used in the nursery, we could find 6 known and 3 new Phytophthora species, while in the irrigation system of the nursery, 21 known Phytophthora species and 6 new Phytophthora species were found. What is the impact of nursery infestations on forests and natural ecosystems in Europe? Between 1990 and 2010, these decades where we have good and reliable data, 6 million hectares were afforested, first afforested, and 22 million hectares were reforested, harvested, and then forested again with plantings. The average Phytophthora infestation rate of the forest nursery stands and plantings of 80% multiplied with these 28 million hectares gives an incredible number of 22.4 million hectares of Phytophthora infested forests planted across Europe in these 2 decades. And since that, certainly many, many more million hectares. The average afforestation size in Europe is 70 hectares. That means 2.6 million infested planting sites were established in these 2 decades. And from every one of these planting sites, Phytophthora are then spreading into the neighboring ecosystems. The volume of forest nursery production and trade in Europe is simply breathtaking. We produce in Europe, in the EU, 2.3 billion forest nursery plants per year, and 30 million of them are traded even between the individual countries. That production differs between the different regions of Europe, while in Northern Europe, only 8.5% of the forest nursery seedlings are bare-rooted. In Eastern Europe and Central Europe, 85 to 88% of the seedlings are produced in fields in the ground with these accumulating Phytophthora species in the fields. And in
Thomas, I think you got muted again. Can you just quickly unmute yourself?
It went automatically. That's very shocking. Can you hear me now?
Yes.
Okay, I'm very sorry. Was this very long?
No, it's just, you know, as I said, I told you as soon as it happened.
Okay, thank you.
You just, yeah.
In the 35 years between 1990 and 2025, according to FAO data, the area of planted forests in Europe increased by 8.1 million hectares. On a global scale, the increase in these 35 years was even 120 million hectares. Considering the Phytophthora infestation rates of nursery plants, these are really shocking numbers. Here I show you the volume and intensity of the EU nursery trade, also including the ornamental nursery trade. It's taken from the study of Dehn and Schmutz et al. 2010. And it shows that it is a very intense nursery trade network all across the EU and Europe. And 1.7 million tons of nursery plants are traded every year, with a few very big players, Netherlands, Germany, France, and Belgium. Where do all these Phytophthora species, the 109 invasive introduced Phytophthora species, come from? We are almost certain that they were all imported with overseas plant imports. In the IC4 project, a European finance project in 2012, they found a number of 4 to 5 billion plants for planting with adhering soil on the root systems were imported per year. 4 to 5 billion plants. And the monetary volume of the plant imports into the EU has exponentially increased in the last 10 years and is now 2.5 billion euros. The new EU Nature Restoration Regulation aims to plant at least 3 billion additional trees until 2030. This is a huge number, and it is more than per year is produced up to now in the EU. How can we ensure Phytophthora-free plant production in nurseries to prevent the nature restorations, the afforestations and the reforestations to make the forests more resilient to climate change, causing a phytosanitary disaster? It's very clear that we urgently need an EU-wide nursery accreditation scheme. There are successful nursery accreditation schemes from Australia, where they have 2 different accreditation schemes that work very well and clean the nursery industry from Phytophthora and other pathogens. They also have 2 different systems in California, and Tyler Bourret will later introduce also this accreditation to improve restoration air in California. And we have in the in the UK since a few years, the Plant Healthy Nursery Accreditation Scheme, which is very successful and well working and could be an example also for the EU. And we will later hear from Malcolm Catlin, the Director of Plant Healthy, about this nursery accreditation scheme. The transition of the EU nursery production system, which is overdue, will be expensive and it will need financial support by the states. That is clear, and the scientists will certainly argue for this with the individual state organizations. Thank you very much, and I hand over to the next speaker.
Thank you, Thomas. Whoever, if you have questions, please remember to state your name, your affiliation, and the speaker you're addressing the question to in your chat. And with that, I would like to ask Bruno, Dr. Bruno Scanu, to take over. Yes?
Sorry.
Can you just start your camera?
Can you hear me?
Yes.
That's all the message. It's difficult. Anyway, I can share my screen. Can you see my screen?
Yes.
Okay. Can you see my presentation?
Perfect.
And presentation mode. Okay, perfect. Thank you, Andrea, for the introduction and for organizing this interesting webinar. Move this, sorry. Otherwise I have. Okay, good afternoon, everyone. In my presentation, I will be talking about the implementation of best management practices to produce Phytophthora free plants in forest nursery. And specifically, I will present a practical case study in Sardinia, Italy. So this is part of a collaborative project between the University of Sassari and the Agris Research Agency in cooperation with the Forest Agency, which is managing the forest public forest nurseries in Sardinia. And this project was funded by the regional governments. And here are all the main people that have been working in this project, and I take the opportunity to acknowledge all of them. Following Thomas' presentation, I also would like to highlight the risk of using infected nursery plants as a major pathway for spreading Phytophthora into natural and forest ecosystems. And here you can see severe dieback caused by Phytophthora cinnamomi on a cork oak plantation in Sardinia, which was planted around 30 years ago through EU program for afforestation of former agricultural lands. And because the economic value of the cork oak was one of the main choice for forestry, and at that time there was a high demand of plants, and we imported many cork oak plants from other countries, and this is probably the way how we introduce Phytophthora cinnamomi. And here are the symptoms of the pathogens. It is basically destroy the fine and lateral roots and can cause also necrotic lesions on the main taproot and exposing the trees to the summer drought in Mediterranean climate. Once introduced Phytophthora, it does not kill only the planted plants, but it can be also spread. It can spread into the natural environment, infecting also native vegetation. Not just forest, but Phytophthora can be introduced also in urban environments and gardens, which are actually even more suitable for Phytophthora infection because of the irrigation, warmer temperature, and other stressors that affect the trees. It is
Bruno, I think you have to unmute yourself. I don't know why sometimes it just switches over.
Okay.
Yeah, you're good now.
Okay, can you hear me? Okay,
yeah.
Good. It is also true that from nurseries, the Phytophthora can be spread also into natural environments, especially in touristic places like Sardinia, where we have villas with big gardens, many exotic plant species brought from overseas that can be introduced, also exotic Phytophthora species. So because of the increase of Phytophthora in forest and natural ecosystems and the link with infected nursery plants, we, together with Agris and Forestas, we started a survey in public forest nurseries to assess the presence and diversity of Phytophthora species and the diseases they cause. Identify the key risk factors for Phytophthora contamination across the production chain. And finally, we selected a nursery where we implemented the best management practice to produce Phytophthora-free plants. So for isolation and identification of Phytophthora species, we firstly sampled disease plants. And we did the isolation using the baiting techniques, which is based on the use of baits like apods, flowers, and leaves. We can also do direct isolation from infected tissues on selective media. And we identify the isolates using analyzing morphological and cultural features, as well as amplifying and sequencing different gene regions, such as ITS, COX-1, and beta-tubulin. We also randomly sampled asymptomatic plants and the different growing substrates that are used in the nurseries, and also the potting media in the different phase of the production chain. We also sampled the irrigation water. We collect some water samples and brought to the lab, or also made some baiting directly in the water reservoir. We also collected the runoff water from the channels, and also the reuse pots, especially those that had soil particles, potentially with Phytophthora spores. And also we took samples from the green waste. So going to the Phytophthora disease symptoms in nurseries, when we go into the nursery, we usually see very nice plants, green, because of the irrigation, the nutrients, and also the use of fungicides that often mask disease symptoms expression. But if we look carefully into these plants, we can start to see some typical symptoms of Phytophthora, such as chlorosis or dieback in the leaves. And if we look at the root system, we usually see root rot or necrotic lesions on the main tap roots. And particularly from these samples, from these plants, which are mainly cork oak and olm oak, we found multiple Phytophthora species, including Phytophthora cinnamomi and Phytophthora quercina, that are usually isolated from oaks, but also other species, new host associations like Phytophthora nicotianae, Phytophthora niederhauseri, and Phytophthora heterospora, which are typically in a nursery environment. In other case, we clearly see the disease symptoms, like in this case, for example, root rot on Pinus pinea seedlings caused by Phytophthora pseudocryptogyia. And it was actually quite an interesting story, because these public nurseries are managed by the same agency. So there was a connection between the nursery. The disease was first found on nursery 1, and then we found the same symptoms in another nurseries when the plants were moved before they showed the symptoms. So this can happen even with different nurseries, not just those managed by the same agency. And what we found also is that Mediterranean shrub species, which actually in our in Sardinia or in Mediterranean countries are often used as ornamental plants in gardens. They were shown to be very susceptible to Phytophthora infections. For example, Myrtus communis, which was severely affected by Phytophthora nicotianae, or Pistachia lentiscus with Phytophthora niederhauseri, or Arbutus unedos infected by Phytophthora parvispora. And also in nursery, we can we found actually aerial infection by Phytophthora, which are quite unusual in Mediterranean climates. But this it's to show that in nurseries, the conditions are very suitable for Phytophthora infection, such as leaf infection and twig infection made by Phytophthora cactorum, Phytophthora citrophtera, on Viburnum tinus and Laurus nobilis. Looking at the Phytophthora diversity that we found in our surveys, we analyzed over 600 samples, and more than 45% were positive to Phytophthora. A total of 58 plant hosts resulted positive to Phytophthora, with cork oak and olm oak showing the highest disease incidence. A total of 37 Phytophthora taxa have been detected. Some of them were very widespread, such as Phytophthora nicotianae, Phytophthora cinnamome, Phytophthora multivora, Heterospora, Phytophthora bilorbang, while others were less rare or rare. So moving to the critical issue that we commonly found in nurseries, a high risk of contamination was the soil and the potting media. Usually this material came from outside the nursery, and we found in some cases this substrate already positive to Phytophthora, to multiple Phytophthora species. In other case, this growing substrate was negative to Phytophthora, but then it was stored in a place where Phytophthora was present, so it was easy to get cross-contamination between different substrates. Another high risk of contamination is also growing the seedlings on the ground, as you can see in this picture, especially if you have an already infested soil in a nursery. If you plant susceptible hosts such as sweet chestnut or walnut in the soil, they can get infested by Phytophthora very easily. And it is also the same if you grow containers or pots in contact with the ground. Also in this case, the roots become easily contaminated by Phytophthora. As shown by Thomas, water is very important for the Phytophthora life cycle. So spending water and flooding in nursery is another high risk of contamination. As you can see here, when the plants are over-irrigated, there are some flooding water, and we can isolate the different Phytophthora species from this water. So situations like this with the poorly drained surface are particularly suitable for Phytophthora proliferation. We also found some positive samples on the irrigation water, so we consider that a possible source of contamination. And that's why we had a different origin of the water. Sometimes it was from wells, other times where was the water, irrigation water, we use river water or lake. And we found the typical aquatic Phytophthora species, such as Phytophthora gonapodides or Phytophthora inundata. Another critical issue we found, and we consider a very high risk of contamination, was the green waste and cool pile. We found situations like this in many nurseries with mountains of probably dead plants because of Phytophthora. And this area were often not well managed, so any movement of this material was a high risk for spreading a Phytophthora into the nursery. Another possible risk of contamination, especially when we reused the containers, if they are dirty with the soil particles, they can even have Phytophthora spores. And even the lack of hygiene measures across the production chain can be a possible source of contamination. And also, when we imported the plants from another nurseries that is not using our growing conditions, even if they are asymptomatic, they can still bring Phytophthora. And especially if in the nursery are using fungicides, fungicide that can mask disease symptoms and also select resistant strains. The high diversity of Phytophthora species that we found in our survey and the high risk of introduction through restoration programs, especially considering the new EU directives on reforestation and nature restorations, which aims to plant 3 billion additional trees by the 2030. The main challenge in the next years will be to meet the demand for healthy plants and reduce new outbreaks. So based on the previous experience and accreditation schemes developed in Australia and California and more recently in the UK, we selected an area which we call it a pilot nursery, in a nursery managed by Forestas, where we implemented the best biosecurity management practice. So one of the first things that we did was to improve the drainage and with a gravel layer to avoid standing water, as we saw how important was to avoid water lodging in the soil. We put a fence around this area, and only trained nursery operator can work in this pilot nursery. Soil and growing substrate were steam sterilized. It was a bit challenging for the nurseries because they had to buy a new steam generator, which is quite expensive, and also was important to ensure to store this substrate in clean areas. And also we introduce regular disinfection of working areas, benches, machines, and tools that were disinfected with 5% of sodium hypochlorite. And where possible, we use new containers to avoid the presence of soil particles with the Phytophthora spores. Otherwise, we properly disinfected the used containers using a 5% sodium hypochlorite. And very important, especially in our case that we were working in a nursery that was already infested by Phytophthora, was to raise containers above ground to preventing standing water and soil contact. And also this was quite challenging for the nursery because these benches are quite expensive. But after 1 year, the nursery, the Forestas agencies start to produce themselves these small benches, which results as very good in preventing Phytophthora infection. And then irrigation water, we know that is also a possible contamination source. In our case, we mainly use deep well water, and we know that this is usually free of Phytophthora. We try to adopt also a filtering system for the irrigation water, but this was a bit complicated. And then we store the irrigation water in disinfected tanks, as you can see here, again with 5% of sodium hypochlorite. We also try to recapture the runoff of water and treat it with approved methods. Very important, we produce plants only from seeds. Phytophthora cannot be moved endophytically inside the seeds. So if they are collected from healthy forest, and then they are planted in a sterilized substrates, it's better to grow plants from seeds. In case we imported plants from other nurseries, they should be applied the same growing conditions. And we also implemented this very strict hygiene measures, and by cleaning, for example, the footwears or all the tools. and implemented phytosanitary monitoring plans. There are many rapid diagnostic kits that can help operators to detect Phytophthora and eliminate the plants, the infected plants. We put a lot of effort on information and staff training and keep them up to date. This was quite challenging because when we started was COVID, the end of the COVID time. So it was difficult to meet the people in person, but it was very worthwhile doing because we realized that nursery operators are very keen in producing healthy plants because they have a reputation and they really want to learn how to implement this pest management practice. And we also did field campaigns. We went into the nurseries with the nursery operators to recognize Phytophthora symptoms, which is not always easy, especially in nurseries. We also created a laboratory in the nursery. We reproduced the Phytophthora life cycles because, as I showed by Thomas, for people working in nursery, it's good to see, for example, the spores of Phytophthora to see what they are actually fighting. So going to the results and the conclusions, I don't have many numbers to show you, but clearly you can see from this graph that within the nursery, the pilot nursery, we were able to reduce significantly the Phytophthora infection compared to the same nursery by using conventional methods for growing plants. And I put this photo of Castanea sativa, chestnut seedlings. Foresters Agency were many years were not able to grow chestnut because of inky disease caused by Phytophthora species. And in our pilot nursery, we demonstrated that they can grow healthy chestnut plants. Another sign of the successful best biosecurity management practice is that they are scaling up this practice in the conventional nurseries and also in other nurseries that are managing, that Foresters is managing. And also we recently had some interest from the private sector to implement these measures. So finally, and concluding, forest nurseries still represent the major pathway for the introduction of exotic and invasive Phytophthora species in Mediterranean forest ecosystems. The current EU directives of urban forestation and nature restoration program underline the importance of producing plants free of pathogens. Best biosecurity management practice reduce the risk of introducing and spreading destructive plant pathogens, protecting forest and natural ecosystems. I will leave you with the message of our webinar, Healthy Nursery Plants, Key to Healthy Horticulture, Forestry and Natural Restoration. The lesson that we learn from our project is that we need to work together with stakeholders, nursery operators, decision makers and scientists together to protect, to produce healthy plants and protect our natural ecosystems. Thank you very much for your attention.
Thank you very much, Bruno. Thank you very much. And while I think Dr. Ana Pérez-Sierra can unmute her microphone and start her camera and share, I just want to remind everybody to keep your questions posted in the chat. Make sure that you write your name and the person you want, your affiliation and the person you want to address in the chat so that we can address your questions during this discussion session. Thank you. Ana Sierra.
Hello, and thank you very much, Andrea. I don't know if you can see my screen.
It's good.
Yeah, okay, then I go. Thank you very much, and hello to everyone. I'm going to talk about the effective and sustainable strategies for Phytophthora management in horticulture. And first, we need to ask ourselves why plant health in nursery matters. I mean, we have seen from Thomas and from Bruno what is the situation of Phytophthora, and we see that nurseries are one of the starting points of the plant production chain. In fact, they supply plant material for agriculture, landscaping, forestry, gardens, And obviously, the objective of the nurseries is to produce strong, high-quality plants that arrive to their destination healthy. And when I mean healthy, I don't mean only asymptomatic. I mean plants that they are free of pests and pathogens. However, sometimes the quality does not depend only of how the plant looks like or the plant growth. Also depends of the phytosanitary status. As we know, nurseries provide the ideal conditions for plant growth, but also they provide the ideal conditions for the development of pests and pathogens. And when a pest or pathogen originates in the nursery, it can spread through and easily to commercial crops, gardens, or natural areas. And they can, in severe cases, cause economic losses. Then nurseries, as we have seen, they play a critical role in preventing the introduction and spread of plant pests and pathogens. But what is the impact of Phytophthora in nurseries? I mean, we have seen a few examples already from Thomas and Bruno, but we have several studies, and I'm going to highlight here one study we did on Phytophthora infestations in Europe, and during or between 1972 and 2013, we participated, 23 countries participated in this study, and we looked to over 700 nurseries and also over 2,500 areas in which trees and shrubs were planted. And to our surprise, as Thomas highlighted earlier, we detected in nurseries infestation rate of 91.5%, and we detected 49 Phytophthora taxa in nursery. Of these 49, 41 were alien, 3 native, and 5 were cryptic taxa. And you can see from the graph here that we have, as mentioned earlier, Pluribora, Cinnamomum, Cactorum, Citrophthora ramorum, Citricola, Cryptogaea, Cambibora. You see all this range of different Phytophtheras, and this little piece of cake here that you can see is another range of Phytophtheras. But this is not the only study. We have more studies. This was in 2016, and studies of the impact of Phytophthora in nurseries, they have been published. And here I highlight a few, some previously, but most of them published in 2024 or 2022 and 2024, then in the last few years, just to show in Spain, in Portugal, Czech Republic, in Italy, the impact of these Phytophthora in nurseries. And I wanted to see what they do in ornamental nurseries, and I'm going to focus on in the Mediterranean area, and especially the results I'm going to show they are from Spain. And these are some of the Phytophtheras that they are very common. And as you will see the names, you already have seen them in the previous presentations. Then we have some of the Phytophtheras will affect the root system, and the plant will die. And here we have on the first case, Phytophthora nicotiana on lavender. The plant will collapse because of the root rot. The same with, we had one case of Phytophthora tentaculata on Santolina. The whole production, it was destroyed. Phytophthora palmivora on Pittosporum, causing the wilting of the plants here. You can see Phytophthora citrophthora on Evonimus, causing dieback. Also Phytophthora niederhauseri on Edera. You can see the wilting of the plants. And Cactorum, Phytophthora cactorum, in this case, instead of affecting the root system, it was affecting the leaves. It was aerial infection, and you can see the black spots caused by this Phytophthora. Also, in nurseries, as we said, you have this whole range of Phytophtheras on all these different range of hosts. Here we have Phytophthora asparagi on agave, and you can see it's a crown rot, and you can see how the infection is going up here. Or Phytophthora drechleri on Scalonia, Phytophthora cinnamomi on Ilex aquifolium. We have Phytophthora cryptogaea on Arbutus or Pistacia, or Phytophthora citricola on Viburnum. In these cases, I'm just mentioning one Phytophthora in one host, but in many cases, we find more than one Phytophthora species in any of these hosts. Then it's quite usual to find, in some cases, you only find one, but in some cases, you find several Phytophthora on the same host. In the Mediterranean area, also the crops are affected by the same Phytophthora. For example, we have the impact on citrus trees. You know, we are, especially here in Spain, we produce a lot of citrus, and we have two main Phytophthora that are always affecting the root system of or the variety of citrus. Even they can affect the fruits. Normally, you will have the dieback of the branches, especially the gommosis. You can see the exudation of gum, not only in the trunk, but also on the higher branches. And also you can get the fruits affected, especially by a splash when these pathogens are in the soil. When we have rain, a splash of water will go to the lower fruits and cause infection as well. If we look to the root system or the base or the roots, we'll see these kind of lesions that you can see here in this picture, or here, these lesions caused by Phytophthora. In severe cases, some of the fields can be severely affected. Another important crop in the Mediterranean is olive. And as we said, sometimes you have your nursery plants, they go to the field, and within short time, you have death of trees. As you can see here, you can see some of the dead trees. When you go to the base of the trees, you can see the foot rot or the crown rot here, and the lesions at the base. In this case, different Phytophthora were involved, one of them being Phytophthora heterospora, is a recently described Phytophthora species, sister to Phytophthora palmivora. Another important crop we have is almond. We produce almond trees, and we found Phytophthora niederhauseri affecting almond a few years ago. And what I want you to see is we have death of trees, a lot of gommosis. You can see at the base of the trees, and lesions on the roots. And the three bottom pictures, they just show the time lapse in 1 month. We went to see the field, we detected Phytophthora niederhauseri. Second was middle of the month. By the end of the month, all trees were dead. Also, we have another important crop for the Mediterranean, pomegranate, and we have Phytophthora palmivora affecting pomegranate. Again, general decline affecting the crown and the roots. You can see here the root systems affected, the lesions on the root. Another important crop, avocado. And here we have the effect of Phytophthora cinnamomi on avocado, decline and death of trees. And now I have selected three of you see these Phytophthora are very common in different environments in the Mediterranean, but I have selected three just to go a little bit deeper into them. Phytophthora nicotiana, this Phytophthora has a broad host range, infects over 250 genera in 90 plant families, and it can cause diseases in agriculture, horticulture, all around the world. And as we saw, we affect citrus, and is one of the most common species in nurseries of potted ornamental and fruit-tree species. And this is very important because the trait of these represent one of the most efficient dissemination pathways for Phytophthora species. And here is just to show that it doesn't only affect these plants, it also in urban trees has been detected recently in areas of Greece. This is Phytophthora niederhauseri. This is an emergent Phytophthora in Mediterranean climate. And these Phytophthora affect the roots and crowns and stems of 33 plant species in 25 unrelated botanical families in 13 countries. This species was described in 2014, and since then it's been described. I highlighted here some of the articles. These are very recently, how this Phytophthora is becoming more important and more relevant in our climate, not only affecting ornamentals or crop, but also recently described also on Eucalyptus globulus. Or Phytophthora cactorum. This is another Phytophthora that can cause from fruit rot in strawberries, food rot or basal rot, stem rot in apple trees. It can be causing aerial lesions on aucuba, or it can be affecting the roots of European beech trees and causing the dieback. What I wanted to show with all of this that I'm telling you is that all these systems, agriculture, horticulture, gardens, park, forestry, natural ecosystems, they all have one thing in common. And these phytophthora are, these systems are interconnected. And what we have in common in these three systems, one is the human activity, the plants, soil, and water. And this is how phytophthora can move amongst all these three systems. Then, this is a diagram that our colleague Marilia prepared for us. And if you have phytophthora coming maybe internationally, ports of entry, or it could be an arrival into a nursery. Normally, these plants arrive, and when they arrive, they are easily dispersed to agriculture or horticulture, to forestry, to natural ecosystems, to public gardens. And all these systems, as I said, they are connected. What we have in common, all the systems, as I said, is plants, soil, water, and the human activity amongst these systems. And this is very important to know how easily we can move these Phytophthora pathogens. Then what are the strategies to manage these problems in horticulture? And I'm going to try to highlight some of them. The first thing we need to take into account is that the management of Phytophthora is not going to be, is complex because the systems we are working are complex as well. Then we have nurseries, and nurseries are very complex systems. Why? Because first we have a very wide range of plants. We have a high plant diversity. We have different plants families, species of cultivars, all in the same space. We could be trees, shrubs, flowering plants, all different plants, all in the same place. They may come even from different origins. Then if they bring any new Phytophthora or any Phytophthora, they are coming all together under the same roof. We may have different cultivation methods, different microenvironments. And if you see only on this point, the high diversity allows for cross-infection to occur, and it may lead, as I mentioned, to new host pathogen combinations, because this is the place where they all meet, plants from different origins, different plants, and maybe different pathogens. We also need to take into account that the plants are at risk to get disease. Why? Because because of the production system, because they are fragile, and because we may have practices that they are not adequate. If we look to the production system in nurseries, what we see is that first we have a highly intensive production systems, dense plantings or production systems, and we do a lot of movement of plants. Plants, they move through different production areas within the same nursery or even between nurseries. What happens is that these provide numerous opportunities to disperse Phytophthora. And the diversity of plant species promotes the cross-infections. It even favors the formation of hybrids, where that in Phytophthora is common. And also in the production system, we have the favorable conditions for Phytophthora, warm temperature and high humidity. As we said also, plants, they can be stressed because they are growing outside of their natural environment, and we are forcing them to grow, and also they may be a bit stressed. And we may have practice that they are not adequate. And I think Bruno has highlighted in forest nurseries, and this is the same in horticultural nurseries. I mean, we have nurseries that where they are using or reusing infested compost, using or recycling infested containers. They recirculate contaminated water. We may have poor drain surfaces. In many cases, it's a direct contact of the plants with infested soil or with surfaces. We may discard the debris or disease material very close to the production area, and that's another focus of infection. And maybe even we have a lot of weeds that they can be a reservoir for Phytophthora. Then this is just a graph from our colleagues, Jennifer and Nick. They published in 2012, and I'm using it because I think they highlight very well how we need to look to the pathways, how Phytophthora can enter our nurseries, and we need to recognize the main routes into the nursery. One of them is the incoming plants. Incoming plants, they might be infected. The substrate, the potting media, the substrate that we use, the mixes that we use, it could be infested. The irrigation water, it could be from water wells, reservoirs, rivers. They may have also Phytophthora. Then we need to take that into account. And also the irrigation system that we use, the type of irrigation system, it can favor the dissemination or dispersal of Phytophthora or water splash. And we need to take action. Also, the containers and trays that we use in the nursery, they need to be disinfested as well. And the growing conditions that in the nursery, we could have different parts of the nursery having different growing conditions. Some maybe in pots, some maybe in the ground. And we need to take action in every single step of the production chain to address the possible entry of these pathogens. We have something even more, some more problems added to this, is that the infection may happen and we may not see why. Phytophthora is microscopic. We are not going to see it. Then sometimes it can be overlooked. Infected plants, sometimes they're asymptomatic. And it's only when they have the right conditions, the symptoms will appear. The pathogen is there, just waiting for the right conditions. If we have used fungicides, we may have suppressed the symptoms, therefore we cannot see it. And also some of the amendments that we use, they can be infested, but it go unnoticed. And it's not an easy, as I said, management of Phytophthora can be complex. But how we reduce the risk? The first thing is prevention. Do not allow Phytophthora enter the nurseries, full stop. That is the first thing. But you know, sometimes you cannot control everything. Then use disease-free plant material, very important. Use non-contaminated soil substrates, mixes for your substrate to grow plants, or disinfect them before use, like Bruno showed earlier. Use clean tools or machinery to prepare container mixes of soil. Use disinfested containers or trays, or use new ones. This is very, very important. Keep containers on well-drained surfaces. Avoid having them standing in free water. Raise the containers off the ground, or maybe add a barrier. You can add a barrier between the container and the ground, just to make sure that we don't have our plants in direct contact with the pathogen. Avoid rotation of crops in the same beds. Store all the tools, machinery, container mixes in clean places. Remove all the plant debris and leaf litter from the soil surface. Disinfect all the tools, footwear, vehicles when you move between different areas, because it's very easy to take Phytophthora. If you have muddy shoes or muddy wheels, you will take Phytophthora within that soil that is attached to the wheels. And also, if you use fungicide, be aware that you may select for resistant strains. Regarding irrigation water, the same. Use a system to make sure that your water is clean, filtering or disinfestation of irrigation water with barriers, filtration, others, any kind of filtration. Avoid the sprinkle irrigation, as we said, because this can flush propagules from the soil surface, from plant debris, or from infected plants. Another point, very, very important, is to treat the runoff water. If you are watering your plants and you have runoff water containing Phytophthora and goes to another place, it may infect these plants, and it's very important to treat the runoff water. All of these measures are very good when you're looking to the plants, to the nursery, ways, the pathways of entry, but this is not enough. We need to train staff. And this is very important. Staff needs to be aware of the problems caused by Phytophthora or other pathogens and the ways of disseminations. It's very important because they work in the nurseries and they need to know and to try to avoid spreading Phytophthora. Also, to know about the diseases and be able to identify early symptoms. Although sometimes if they are symptomatic, it's quite difficult. If a plant dies, don't ignore it. Always diagnose what is wrong with a plant, because that could be the beginning of a new infection. Diagnose the problem promptly, and when you identify the pathogen, it may be Phytophthora or something different, you can apply the control measures accordingly. Then just to conclude, the effective plant management in nurseries relies on prevention. Prevention and knowing how it can come in and reduce the risk of Phytophthora coming into our facilities. Then the management of Phytophthora include good biosecurity measures and good practices in the nursery, training staff, very important monitoring and surveillance. Keep an eye always on your plants, make sure that you don't miss an early infection. Early detection is key, a rapid diagnostic method, and that will allow you to apply effective control measures. Then these biosecurity measures and good practices are the key to minimize the risk of introduction of and spread of Phytophthora. But we know, and it's been highlighted already, we need better protection systems. We need to improve in surveillance and monitoring. We need to address any gaps in national phytosanitary regulation and legislation. And the most important one is that we need to work together, as my colleague Bruno highlighted earlier on. And I leave it here. Thank you very much for your attention.
Thank you very much. I see that the questions in the chat are accumulating. This is really nice. We will wait with answering these questions until the discussion time. So, keep it there. Please do not use the chat for promoting yourself. This chat is really just for the questions regarding this webinar. We are recording this webinar. So for people that are not here, that would be a choice, and we will also post it and send you the link. For now, we have a short break now for 10 minutes. So please come back at 15:35, I think, 25, hold on, 25, and then we will continue our webinar with Dr. Sarah Green. So I hope to see you all back then. I will mute my microphone and turn off the camera, and then we will start again in about 10 minutes. See you then. Right, I think it is time to start again. So with that, I would like to, I hope you can hear me all. Good, well, okay, I would now like.
I can hear you.
You can hear me okay?
Yeah, yeah.
Excellent, good, clear and green. Dr. Sarah Green, if you would, I see that your presentation is already there, so I will give you the floor to talk about Phytophthora and British plant nurseries.
Okay, thank you very much. I'm assuming you can all see the screen and it's in full mode.
Yes.
Yeah, well, so thank you. Yes. I'm going to talk about Phytophthora and British plant nurseries now, identifying risk and which best practices are best. Before I start, I'd like to acknowledge all my colleagues and collaborators who worked with us on this project over the last few years from several institutions in Britain. So I just want to give you an overview of what I'm going to present. I'm going to start with explaining how we did this nursery sampling for Phytophthora. We did it as part of two projects. One was the Phytothreats project, which was only in Britain, where we sampled nurseries just in Britain, and that ran from 2016 to 2019. It was funded by sort of UK research councils and sort of devolved administrations. We also did nursery sampling as part of a UFresco funded project, IVIA, and that ran from 2020 to 2022. That involved a much broader number of partners from, so nursery sampling was done in numerous countries, not just in Europe, but also in America and other places. But I'm only going to present the data from the British nurseries from the IVIA project. So I'm then going to talk about how our Phytophthora findings relate to nursery management practice and which best practices are best in terms of prioritizing what to do first, if you like. And then I'm going to finish off by explaining how we disseminated our information to growers, but also how we also engaged with the Plant Healthy Accreditation Scheme, which was developing at the same time that we were doing our research. So in terms of nursery sampling for Phytophthora, we did what we called a fine scale survey of 17 different British plant nurseries between 2016 and 2022. And then these roots and water. And these nurseries, we had 10 nurseries in Scotland, 6 in England and 1 in Wales. So they encompassed a broad area of Britain. And the majority of nurseries were wholesale ornamental plant nurseries, where they dealt with or traded in herbaceous perennials, shrubs and trees. We also had 5 forest tree nurseries and we had 2 specialist nurseries. So again, the nurseries were quite broad in terms of their practices. So for this fine scale survey, we did 2 to 5 visits per nursery over 2 to 3 years. And in addition to this, we carried out what we called a broad scale survey of 118 more growers, where roots only were sampled as part of annual statutory plant health inspections. We carried out a standardized set of methods for sampling, which I'll explain in brief shortly. And we identified our Phytophthora pathogens primarily using a method called DNA metabarcoding, which again, I'm going to go through very briefly. And the detail of our methods and of our results have all been published in this paper, which was published in early 2025. So in terms of root sampling, we sampled roots from fine roots, that is from stock plants and from plants that had been placed in plant dumps. We collected roots from 3 replicate batches of plants per host sample, and we placed those into pre-labeled envelopes and they were freeze dried. And then in terms of water sampling, we collected water samples from any water source that was used for irrigation, and this did vary widely. Some nurseries used what we call closed water sources, so they were mains water or borehole water, and others used open water sources, for example, in the top picture here, open reservoirs, some extracted directly from rivers and some supplemented with rainwater. In many cases, the open water sources were not treated to kill pathogens, so it was untreated open water sources for some of our nurseries. We also sampled water from puddles and runoff from plant stock and waste piles. And if there was a stream running through the nursery or alongside the nursery, we also sampled water from that. So just to illustrate what we did for our water sampling, it was David Cook gallantly illustrating our knapsack sprayer technique. Essentially, we took 5-litre samples of water and we put them in a knapsack sprayer. You can see David's got on his back, and then the water sample is pumped through a filter. And in the top right-hand photo, the end of the sprayer has been modified so that we can put a cellulose filter. So the idea is that any Phytophthora propagules, so zoospores and any other propagules, collect on this filter. And then we run 3 replicate 5-litre water samples per sample. So we have these replicates, and the filters are placed in a buffer solution. We also carried out what we called water flow through sampling, where we placed batches of plants in trays, and we had 3 replicate trays per batch of host. And I wanted to, the photograph here was from Greece. It's from Demosthenes, who sent us this photograph. This was taken as part of our IDFIGHT project. It was the best photograph that I have to illustrate the method, and I want to emphasize I'm not presenting any results from Greece. But it shows how the plants were watered from above. The water percolates down through the pot, through the roots and out into the bottom of the tray. So the idea again is that any Phytophthora propagules in the roots or in the potting mix or even on the foliage are run through, washed through and collect into the bottom of the tray. And we left these sitting for at least 30 minutes, and then we filtered the collected water as I previously described. Brief run through our DNA metabarcoding method. Essentially, we extracted the DNA from the freeze-dried root and from the buffer in which the filters were placed, because largely the Phytophthora propagules would wash off the filters and into the buffer. And we carried out a nested PCR that aimed to amplify Phytophthora DNA. And again, thank David Cook for this schematic on the slide here, that for anyone who's interested in the method, it shows you this molecular technique that will amplify, if a Phytophthora is present, it will amplify that sequence or the sequences that are there. So that's the first stage in the testing process. So all samples went through this PCR process, and anything that was positive, which means it has some Phytophthora DNA, those samples were then processed for what we call Illumina sequencing. So Illumina producing, vast numbers of sequence reads per sample, which allows us to identify pretty much as many Phytophthora species present in the sample as there are, rather than just identifying the most abundant or the fastest growing species. And then those sequence data were analysed with this classifier that was developed as part of our phytothreats project. And this was published in 2023. And it's very much based, because we're working with nurseries, we want to reduce the probability of false positives. So we have very stringent quality control, not only in the pipeline, but also through our laboratory processing, so that we can identify and deal with any potential cross-contamination. And then the Illumina sequences are matched to a reference database of known Phytophthora sequences. So you've got that barcode for each species, and the sequences are matched to identify what Phytophtheras are there in the sample. For 4 of the nurseries, we did baiting, which has already been described here. And baiting essentially runs on the principle that Phytophthora spread through free-swimming zoospores, that they can swim through water and they're attracted to the bait plant, and then they form lesions, and then you can plate it onto media and identify through sequencing. But returning to our metabarcoding results, over our 17 partner nurseries, we collected more than 3,000 samples. That was including replicates. We about 150 different host plant species were sampled in this work, and overall about 40% of the samples were found to contain Phytophthora DNA. We detected 85 different Phytophthora species or complexes. But what I really want to focus on here and now is the variation that we observed across the nurseries. So if you look at the figure on the right, what you see is our nurseries are anonymized, 17 of them. So it's N001 all the way through to N017. And it shows you the number of detections of the top 20 most abundant Phytophthora in the study as a whole per nursery. And I think you can see the variation. So for example, nursery N003 and N008 have very much fewer Phytophthora detections than some of the other nurseries. So what I want to talk about now is briefly, how did our Phytophthora findings then relate to nursery management practice? So I don't have much time, and I'm just going to give, again, a very brief overview of the main results. But what we did was a statistical analysis of our data, and this was done by Louise Barwell at the Centre for Ecology and Hydrology. And she used Bayesian stats, looking at the number of detections of Phytophthora species in relation to nursery management practices. And she modelled a set of nursery management criteria that were complete for all nurseries, but which showed variation among nurseries. So on the right-hand side, I will show you the attributes that were actually put into the models and the units that were used in the models for each attribute. So we looked at nursery size, latitude, turnover, and spend on buying in plants. In terms of the management practices, we looked at whether they used an open or closed irrigation source, whether they treated the water before use, if it was open or closed. Did they use their own growing media mix? Did they mix their own, or did they use a commercial compost? Did they reuse pots between batches with or without disinfection? Did they use raised plants to keep plants off the ground? Did they have disinfection mats or stations in the nursery? And then also we looked at, did the nurseries import whole plants from overseas? And if so, did they have a quarantine holding area for those imports? And then finally, we looked at whether waste plant and organic material was dumped on site or not. So in brief, the highest risk nurseries, and that's all I'm going to speak about, these were smaller, lower turnover ornamental nurseries that used open and untreated water supplies, and they imported live plants. And these highest risk nurseries had more diverse Phytophthora communities. They were more likely to have recently invasive species that you've heard about already in this webinar, for example, Phytophthora ostracegae, which is killing our native junipers in the north of Britain, and another recent invasive Phytophthora lateralis. And the photograph I'm showing here is one of those nurseries with the shelter belt of Chamaecyparis lawsoniana that's actually infected with Phytophthora lateralis. And these high-risk nurseries also were more likely to have rare Phytophthora species that were not yet widespread in the UK environment. So I think that's giving you an illustration of how practice can potentially affect the diversity and risk of Phytophthora on a nursery. Louise also modelled looking at what were the highest risk hosts across our study. So this graph is showing you the highest risk genera on the left-hand side, and on the top you've got those Phytophthora species that were found in those plant genera, and the warmer the square is the higher the number of detections of that species on that host. But what we found very fascinating by this was, unexpectedly, Pseudotsuga mensesii, which is Douglas fir, was actually the highest risk host in our study across all genera in regard to the number of Phytophthora species harboured. And so you can see quite a few tree species in this list with Chamaecyparis, Fagus, Prunus, Taxus, etc. And you can run down through. But this was enable us to identify what host genera are most likely to harbour higher diversity of Phytophthora. So I'm now going to move on to, based on our analyses and the evidence and the experience that we had of going repeatedly to these nurseries, which best practices might be best to prioritise in terms of what should you do first, particularly if you don't have huge funding to make big infrastructure changes. And the one thing that we felt would make the biggest difference, and at very low cost, is being much more observant of disease symptoms in a nursery. And it may sound like something pretty obvious, but what we had a lot of evidence for is a lack of symptom awareness. And I want to show you a couple of photos to illustrate this. Photograph on the left shows rhododendron and a nursery that was heavily infected. In fact, the whole batch was quite heavily infected with Phytophthora ramorum, which is killing our larches in the UK. And on the right-hand side, we've got these twisted Leylandii that had recently been imported from the Netherlands with these brown patches of foliage that turned out to be Phytophthora ostracedrae, which is killing our junipers. And what I want to say is that the nursery managers and the staff simply hadn't noticed these symptoms. And either that, or they had noticed the symptoms, but they weren't necessarily linking the symptoms to disease. They were assuming that any browning or drying of foliage could be due to the fact that the plants had dried out, or perhaps they got frosted, or some other reason. We had evidence, we saw sick plants being loaded onto trucks and being dispatched to orders. And so therefore we have these sick plants being planted in the wider environment. So being observant of symptoms is very key. We've heard a lot already about water management, and certainly our data showed that open water sources did contain Phytophthora, much more likely to contain Phytophthora than closed water sources. But also, even those nurseries that use closed water sources, we were finding sometimes Phytophthora in the water, and we put that down to the dirty hose ends. So even if you're spending money on borehole water or mains water, if you've got your hose end on the ground picking up Phytophthora, you know, that's a risk. So we were recommending obviously to hang up those hose ends. As already spoken about, overwatering your plants so that you've got pooling and runoff is also risky, because we found that puddles contain a very high diversity of Phytophthora. In some cases, up to 10 different Phytophthora species in a single puddle sample, including a number of aggressive Phytophoras of woody hosts. And then we've got to go back to these waste piles, and this was something that particularly interested me. So we actually had an additional study where we had an honours student who did a study looking at waste piles on 3 nurseries, and she found in these waste piles 18 different Phytophthora species, but she did some baiting from the waste, and she baited live 8 different Phytophoras. But what was quite worrying was that Phytophthora ramorum, that quarantine regulated pathogen in the UK, was actually baited from the waste piles at 2 of the 3 nurseries. And often these waste piles are located close to hedgerows and streams. And in the case of this one site here, you can see the water collecting at the bottom of the waste pile actually drained into a stream and then run down, you know, into the wider environment. And that was where Phytophthora ramorum was detected. So you have a direct link between a waste pile and the potential spread of an invasive pathogen into the wider environment. And this study was actually published back in 2023 on the high risk of nursery green waste. So just to summarise for this part of it, which best practices are best? Well, we believe having knowledge of those high-risk hosts is important, because a nursery manager can then decide whether to trade in that host or not. Awareness of symptoms is extremely important, and also knowing then what the processes are for dealing with sick plants in your own country. So knowing who the diagnostic authorities are, or what your plant health regulatory bodies advise you to do. That's very important, of having that downstream process for dealing with sick plants properly. Also using clean irrigation water, preventing over puddling, and then managing waste to kill pathogens. Not every nursery has the ability to, or the infrastructure in place to have effective composting, but there are other off-site ways of dealing with nursery waste. And so finally, how did we then disseminate our information to the growers? We produced a series of best practice flyers. They were short flyers. One of them is dedicated to the issue of waste management, nursery waste management, and it outlines what the risks are, as well as what the options are for alternatives to dumping your waste on site, and an idea of the cost of those options. Obviously, this is very country specific. We also produced a flyer that was generally a more general flyer on reducing your Phytophthora risk in nurseries, and then one on identifying symptoms of hill health in plants, and identifying or understanding high-risk hosts for Phytophthora. I should add that all these flyers are available on the Plant Healthy Certification Scheme website, which I think Malcolm will be talking about shortly. And we did work quite closely with the scheme as it was developing as part of this project. So I felt that the science that we were doing was helping to provide the evidence for the plant healthy standard that the nurseries have to adhere to. We held plant health training days in England and in Scotland, which were well attended, and where again we presented the data from our work and how those data support the making changes to management practice. And then finally, as a result of our partner nurseries engaging with us and engaging with the science, we could see how they were actually changing their practices. So, for example, one of our nursery managers, when we were able to report back to him that he was...
Maar ze zou de rest nu ook overmaken.
Could you please mute, please, while I just finish? Yeah, so one of our nursery managers actually installed a borehole when we found that he was watering his plants with Phytophthora from an open reservoir. Other nurseries were started raising plants off the ground, so you don't get that root connection with the soil. Others training their staff in plant biosecurity, particularly around symptom observation and how to deal with sick plants. And then 8 of our 17 nursery partners have actually joined the Plant Healthy Accreditation Scheme, which you'll be hearing about later. And with that, I'd like to thank you for listening.
Thank you very much, Sarah. I think we can just go further on to our next 2 speakers, basically. That's Tyler and Susan. If Tyler, maybe start. Yes, unmute, start your presentation, and you can also start your camera.
And please do all participants mute their microphones, please.
Tyler, we cannot hear you now, so if you muted yourself, just unmute yourself.
Muted myself. Okay. I think everybody can hear me and see me.
Yes, you just have to put yourself into presentation mode.
All right. Thanks, everybody. On behalf of myself and my co-presenter, Susan Frankel, thanks so much for coming. I'm really pleased with the great attendance, and thanks for the invite and especially for organizing this. And so I'm going to talk about this issue of Phytophthora in the context of the US state of California. And really, there's 2 big Phytophthora problems, issues that California has dealt with, and that's sudden oak death, which you've already heard about from Thomas's presentation, as well as this issue of restoration and Phytophthora. And then I'm going to talk about this accreditation program for nurseries that you actually also have heard a little bit about. That's kind of a pilot program to deal with the second issue, the restoration issue. So California has an incredible diversity of all sorts of organisms, but especially when it comes to trees, California has these superlative trees. It has the tallest tree in the world, coast redwood, the largest tree in the world by volume, giant sequoia, the oldest tree in the world, bristlecone pine. It's also got the largest agricultural industry in the US, the largest horticultural industry in the US, which both of them grow a wide diversity of crops, and it's the most a populous state in the country. And so there's just a huge space for all of these plants and all these people to interact, oftentimes in many very positive ways. It's one of the reasons people love California, but also there's some opportunities for negative interactions. So
please
please, please
ऊपर से लेकर नीचे तक सारे पे तीनों उंगलियां स्लाइड करें।
Can you please mute the phone, the microphone?
So Californians.
Maybe Andrea, you mute every participant again, and then Tyler is unmuting himself.
I think we're okay. Well,
Yes.
anyways, anyone who's ever worked for the federal government, this is a known problem with Microsoft Teams. But I'll just move on. Californians live with the effects of sudden oak death every day, especially in northern and central California, with sudden oak death, with the consequences of the nursery pathway. And this is a disease that's killed, tens of millions of trees. We know that it escaped from the nursery industry into the forests, and we now know that it got to North America from Asia. We don't know exactly how, but presumably from the plant trade. And here you can see a nursery growing right in the middle of a susceptible forest for sudden oak death, and we know that's how it got out from the nurseries directly into the forests. Besides all of the ecological consequences and socioeconomic consequences of sudden oak death, there's been a huge amount of consequences and disruption for the U.S. nursery industry because of the regulated status of Phytophthora ramorum. And sudden oak death, Phytophthora ramorum is not the only Phytophthora species that's threatening native plants in California. California has a really incredible amount of endemism, plants that are only found there and sometimes only in a few places. And one of those is Ione manzanita, and this is a plant that's threatened by the biological bulldozer, Phytophthora cinnamomi. So moving on to the restoration side of things in California, there's a ton of ecological restoration in California for sort of the most altruistic reasons and maybe more economic reasons. But in forest, all of the fire that's common in California requires a huge amount of need for new trees to replant forests. And then a lot of the landowners, you're going to hear about a lot of public utility companies in this talk, do what's called mitigative restoration. Laws force them to do restoration as a result of harm that they have to do to the land, disturbance they are doing as part of their activities. And so this problem of Phytophthora and restoration was identified about a little more than 10 years ago in California. Phytophthora tentaculata and California monkey flower is sort of one of the original issues, but it really quickly became recognized as a larger problem. And in California, especially around the Bay Area, there were some really large-scale restoration projects that were ongoing. And in fact, it was quickly discovered that the restoration activities were actually facilitating the transfer of Phytophthora between nurseries, especially through sharing pots after the restoration projects were done that weren't being disinfected. And it's just, these problems were really major. And in California, any land is really valuable. And so despite being heavily infested with Phytophthora, this was land that people couldn't really give up on. I'm a plant pathologist myself. I got involved with this doing a survey as a PhD student at UC Davis. And here we did a survey across the southern San Francisco Bay Area. This is better known as Silicon Valley. So the next time you check your phone, you can think about Phytophthora. And just like many of the other talks you've heard, we found Phytophthora at most of the sites that had been where restoration had been conducted. Later, as a postdoc, I was involved with another project down in Southern California around Los Angeles in Angeles National Forest. And here there was mitigative restoration because of natural gas pipelines and power transmission lines that were put through the forest. The restoration used infected plant stock, and this is an ongoing project where we're sort of monitoring these infestations in pretty arid sites. That also led to a grad student doing some research in that similar area, very arid area, but he was able to find quite a lot of Phytophthora. Just from the diagnostic side, as somebody who is a Phytophthora researcher, one of the things that was part of this process of figuring out what's the scope of the problem was making a list of Phytophthora, and that was something that I participated in. And one of the sort of side pieces of data we could garner from all of this was when I created this list, I sort of pooled all the Phytophthora I could find into these different land use categories. And then if we take the Phytophthora communities we found in these categories and compare them against each other, you can see that the least similar Phytophthora communities in California are the ones in agriculture and the ones in forests. But unfortunately, the two most similar pairings are mimicking this pathway that we worry about. And that's restoration plantings and in horticulture, and then restoration outplantings in forests. So what do we do about all of these infestations and this overall problem? And it's been great working with these public utility groups because they really have more resources than the average entity, and it's their job to sort of manage this land in a long-term holistic way. So on the left there, you see some solarization panels, and those they're placed over a basin that was had an affected plant in it previously, and if you get enough sun, enough time, that can eradicate Phytophthora. On the right, you have a steam auger that can bring the soil up at a particular place and then also treat it with steam to eradicate Phytophthora. Here, another one of these groups is doing a large-scale steaming of some soil. Steam is definitely, I think, a big technology that's emerging for treating soil in this context as well as agriculture in California. Another incredible example on the nursery side of things that's really been the shining light throughout all of this issue is the Presidio Native Plant Nursery. And so the Presidio is this incredible sort of park natural area in the corner of San Francisco. It used to be a military area. You can see it's where the Golden Gate Bridge connects to the city. And they have a great need for native plants for restoration purposes, some ornamentals because it's a big park. And when this issue came to, became recognized, they were able to really put a lot of resources towards creating this nursery that is really a shining example of how it should be done. And it's a public-facing nursery. You can go visit it during most daytime hours. It's mostly staffed by volunteers. All of the phytosanitary best management practices that they've implemented are kind of done in a fun way to the extent that that is possible and are explained why they're doing it. And then even better, this has been around long enough that there are restoration outplantings in the Presidio that you can go visit that were planted with these cleanly produced plants, and they're really successful. And it's really, an incredible example for the public and plant pathologists alike why this, starting with clean plants is so important. So I really want to highlight this working group, the CalPhytos working group, which is sort of an outgrowth of the California Oak Mortality Task Force, which had been started to deal with sudden oak death. And this was a multidisciplinary working group that really sorted through some incredibly difficult problems, figured out what are the problems, working on what were the solutions, and even more incredibly, actually started implementing some of these solutions. There's a lot of resources on this webpage. One of the ones I want to highlight more on the diagnostic side of things, if you're trying to sample for Phytophthora, is this great technical guide for sampling Phytophthora and restoration. And of course, you've already heard from some of the absolute best Phytophthora catchers in the world today. But one thing, if you are doing your Phytophthora survey in the context of restoration, of land management, you're working with land managers, there are some sampling strategies you can add that sort of allow you to do all the science and find all the Phytophthora you're looking for, but also answer some really interesting questions and salient questions for whoever's in charge of the long-term, that land in the long term. Really, there's a bunch of incredible resources for the nursery side of things and the restoration side of things on the CalPhytos website. One of the incredible tools, resources that has been developed by Phytosphere Research through this great working group is this leachate effluent baiting test. And you can see we've got some container plants on a bench and a tarp underneath them, and the effluent lots of water is going to be poured through them. And in that little vessel at the bottom, there's a green pear. And part of making this tool work is the California Department of Food and Ag Plant Diagnostic Lab allows people, including nursery managers, to send those pears in, and they can tell them if they have Phytophthora and if they're interested, which Phytophthora they have. And that's a really incredible tool that allows nursery managers to test for Phytophthora without a plant pathologist coming there to do it themselves. And really, the culmination of all of this is this AIR program. And that's being managed through the new extension professorship at UC Davis, Del Castillo Lab. And it's been around for a few years now, but really already a lot of success. So right now, there's 24 nurseries in California, 10 are accredited, and the rest are working on it. Some of the early findings is that it works best with smaller nurseries, and that it's harder for larger nurseries. Maybe parts of them will be accredited. That makes sense to anyone working on IPM strategies or any microbiologist. But the BMPs, the phytosanitary BMPs are working. Phytophthora hasn't been detected in any of the accredited nurseries. The issue really, as you've heard from Sarah and some other speakers, is that these BMPs can be, quite expensive, time-consuming to get going. Getting good soil treatment is one of the more difficult problems right now. As you heard from Bruno, getting everything up onto benches, because benches are really expensive. And then that leachate baiting test itself is quite labor intensive. It also uses a lot of water, which is not completely abundant in California. And then a few more observations is that even the nurseries that may not go all the way through or be accredited, they're taking a lot of the BMPs. They do want to, produce plants as cleanly as possible, even if some of the BMPs required for the accreditation are a little bit much or a little bit expensive for them to adopt. And this is really one of the issues and that we need to solve as a group, which is that these BMPs make each individual plant a lot more expensive. And if, to the extent there is a market for these native plants, a restoration practitioner whose job it is to just get as many plants as possible may not want to spend a lot more per plant. And so working with these agencies who have the resources to support this and also have the long-term vision why Phytophthora infestation is actually more expensive than clean but expensive plants. So with that, lots of great, so many different people have participated in this, and I thank all of them. And with that, I will wrap up my talk. Thank you.
Thank you very much, Tyler. Again, the questions that you're posting in the chat will be answered in the discussion. So great if you can not only add your name, but also your affiliation and the person you're addressing to. And with that, I think maybe, yes, Malcolm is starting his presentation. The floor is yours, Malcolm.
Thank you very much. And if I can just manage to sort the technology here, I'll put it into slide presenter mode. I don't know why it's not letting me at the moment, but just bear with me. Oh dear, let me just stop sharing for 1 second.
Yep.
We'll have a second go. Hopefully you have that now in full screen, yeah?
This looks beautiful.
Oh, it's fine.
Thank you very much. Good afternoon. I'm Malcolm Catlin and I run Plant Healthy Limited, which is an organisation set up to promote and support good plant biosecurity. Uniquely amongst the presenters today, I'm not a plant health scientist. I come to this job from a background of 30 plus years working in the horticulture industry, and above all with a real belief that the vast majority of people working in this trade want to do the right thing, but don't always find it easy to access what the right thing is. This slide really contains stuff which will be familiar to everybody, but I start every presentation with this. It just sets the context again. The huge range of different types of pests and pathogens that we're up against, the increase in global trade and travel, providing more entry pathways, the use of fewer chemicals through availability and acceptability of use, climate change, which makes it easier, of course, for new pests to establish, and also people looking to more new plant species to help combat climate change and in turn more sources of plants. You don't need me to list some of the specific threats, but we've obviously got polyphagous ones like Xylella, we've got host specific ones, we've got pests that affect ornamental plants, edible crops and so on. And again, you don't need me to remind you of the environmental and the economic and biodiversity and social benefits of trees and plants, and therefore the consequences of getting this wrong. It can be hugely expensive environmentally, reputationally and financially, and of course it's a constantly changing picture, new threats emerging all the time. And this is a slide that I often pop in. I suspect this won't be a surprise either, but this is one showing the rise in the number of serious tree pests of concern in Great Britain. You can see a number of things coming onto the graph in the last 3 decades of the previous century, and then look what happens in the 2 decades since then. The statistic is that the number of serious tree pests seems to be doubling roughly every 11 years, is certainly in the context of Great Britain. So what is Plant Healthy? Well, a very brief bit of history. It was it came about in 2018. Then Prince of Wales, Prince Charles, convened a meeting of bodies in Great Britain, a wide range of trade and professional bodies, and I'll put the slide up in a moment. Bodies with quite different memberships and areas of interest, but with a shared concern to protect the environment, to protect the trade, and so on. And they in turn commissioned the writing of something called the Plant Health Management Standard, a systems-based and proactive approach to minimise the risk of introducing and spreading harmful pests and diseases. And I labour those words deliberately. As I say, independently set up and reviewed, it is still reviewed regularly by a technical advisory group. The Plant Health Alliance wasn't a one-off meeting. That body still meets in full every 6 months. And we have a working group that meets every month to discuss ways in which we can keep up to date and spread the word and so on. Those are the bodies in the alliance in Great Britain, and they cover the tree sector, landscaping, ornamental growing, government bodies, farming, and so on. And although that is a UK body, it is a model that is potentially applicable elsewhere. So a quick run through of what's in the Plant Health Management Standard. It only relates to biosecurity. It doesn't drift off into other areas. It currently contains 32 points, and they broadly come under these headings, which I will rattle through quickly. It makes sure, first of all, that you are meeting the requirements of legislation in the country that you're in, whether that be passporting, phytosanitary certificates, legislation around the movement of wood, forest reproductive material, and so on. It then requires you to have a very clear policy for biosecurity that's specific to your organisation, that sets out what your approach is, and clear, named, specific responsibilities for anything that may impact on plant health. So that's down to which species, which genera you're going to grow or trade, who is responsible for checking plants on arrival, checking them through the growing process and on dispatch, who selects which suppliers you're going to use, and so on. At the heart of it is something called the Site and Operations Pest Risk Analysis, which I'll come on to in the next slide. It also looks at your supply chain, your suppliers of anything that may have an impact on plant health. So obviously any plant material, but also things such as growing media or any wood, whether that be pallets or stakes or wooden crates or whatever that may have an impact on plant health. How have you chosen those suppliers? Who's assessed them? Do they know what they're looking for? Are they trained and up to date, and so on? Plant health procedures looks particularly at nursery practice or practice in any business that's engaged with moving or trading plants, and really links into the kinds of areas that previous speakers have mentioned. So your water source, your disposal of green waste, your storage and selection of growing media, visitors coming onto the site, the use of tools, contractors, anything that may represent a pathway, anything that any stage at which there is a critical check to be done. And it then requires you to have clear procedures in place for all of those, clear responsibilities, a clear understanding of what's being looked for, and continuous records of what has been done. So that checking, monitoring and traceability carries on throughout the process. And of course, all of that requires staff that have been trained in what to look for. So having identified the risks, how do they identify them? Who do they report to if they find a problem? Who do they consult if they're not sure what it is? And so on. Keeping up to date with what, as I've already mentioned, is a continuously changing procedure, and also continuously improving your nursery practices. It's not prescriptive in terms of the methods, in other words, what type of treatment you apply if you have a problem, but it is the framework for considering all of these aspects, and to get advice where needed, to put clear responsibilities in place, and so on. So that systems-based approach, as I said, is very much based around the site and operations pest risk analysis, which is a matrix looking at your host plants and materials, identifying the serious pest risks associated with those hosts, mapping out the pathways, and then looking at the steps that you can take to mitigate those risks and manage those risks down to a minimum level. Or if you can't, to not do those things, and following that to monitor them on an ongoing basis. But it's looking at that whole pathway through the business, looking at your supply chain, how it fits in with your legal requirements, the continuous monitoring, excuse me, looking at the impact of the wider environment on your business, your impact on the wider environment, and so on. We offer certification against the plant health management standard through something called the Plant Healthy Certification Scheme. And that's done through independent annual audits. Excuse me 1 second. We provide a logo, which is a sign of a recognition of that high standard of biosecurity. And we maintain a register of certified members. We currently have 108 members plus 12 applicants. And some of the larger producers in Great Britain are included in that. So we currently cover the production of something like 200 million plants and trees. But we've got a long way to go. And then all of that is run by Plant Healthy Limited, which owns the standard and the logo and administers that certification scheme. Of those 108 members, one of them is now a non-Great Britain member. We've just added our first non-British member, a tree grower in France. And that's critical because obviously this is an international, not a national problem. So why do we need it? We've got statutory measures. People often say that to us. Well, those government inspections only look at samples. They don't see everything, and they tend to focus on what is already on the move or what has already happened. Whereas Plant Healthy is proactive. Its whole aim is to avoid expensive problems and put that good practice into place. It covers everything. It uses the expertise, the eyes of those people who are growing plants, trading plants on a daily basis. As I've mentioned, it's a promotional tool. It's a good way of keeping up to date. And we're also working on a possible insurance scheme for certified members, which would be a bit of a game changer if we can manage to pull that one off, and we are hopeful. Aren't we doing this already? Well, other standards, other certifications may cover some aspects of the plant health management standard, but a lot of it, particularly the critical pest risk analysis and supply chain analysis, are not covered. Growers may say, well, look, I'm doing a lot of this already, but they're typically not doing all of these things. Again, not having that systematic risk analysis in place, not recording all of these things and doing it in that systematic way. In order to have an effective approach and to be proactive, it requires this full systems-based approach, and the standard has been developed and tested on this basis. Who is it for? Well, it's for anybody growing or handling plants and trees, whether they be growers, landscapers, garden designers who are selecting and sourcing plants, those engaged in tree care, those buying and selling plants, and even people like large botanic gardens, of whom we have some, like Kew, in our scheme already. It can be anywhere. So although our membership is mainly in Great Britain, the standard has been written in such a way that it can be applied in any country. And it applies to any types of plants. They carry plant health risks. The same logic, the same proactive measures can apply to trees, amenity plants, ornamentals, edible crops, young plants, cut flowers, you name it. So how might it fit in with other certification schemes, and what view do we take of those? Well, as I've said, other schemes don't tend to cover biosecurity in any detail, and I'm thinking of some of the certifications that are particularly widespread in Europe that cover many aspects of ethical and environmental standards, but typically don't cover biosecurity in any detail. That said, the principles of certification, record keeping, traceability, the basics of good nursery practice, the idea of having an audit will be familiar to existing members of those schemes. And we believe that as our standard is quite short, it will be relatively easy to add that into those other standards. And that way, rather than requiring people to have a separate audit, you can combine them on the same day using existing certification bodies. Of course, not everybody wants to go to the lengths of those standards, in which case Plant Healthy on its own may be the best option. And we're working on the provision of certification bodies in other countries to aid that process. And then, even if you're certified through a benchmark scheme, you would still have access then to the logo and the directory. So why now? Growers have got an awful lot else on their plate, lots of pressures from all kinds of directions. But as we know, the number and level of threats continues to rise. We would argue that it's good to act before the next big problem and not after the event. We see this as being a ready-made tool, a tried and tested tool for a defined issue. Let's just tick this one off the list and take a lead. So what are our challenges? Our challenges are that we've got a strong membership base, a business plan, we've got some funding in place, a small team of people, a good technical base, and the audit systems. But we need to really expand that membership, not just in numbers, but in getting more scopes of businesses, whether they be growers, landscapers, retailers, any of those people, and particularly to improve the geographical coverage. It all starts with awareness, and we would argue that everybody should start by being aware of these issues, aware of the threats, aware of the preventative measures that can be taken, and this is still not sufficiently widespread enough. There's also a lack of public awareness, what they can do to buy responsibly, and indeed what we mean by plant health in this context. So we've got the vehicle in place, but we need to grow it. We need to continue to raise awareness. We then need to get as many people to act, to adopt these principles, and where it's relevant, where it's applicable, to get people to demonstrate that good practice through certification. So our plans very much around building the membership, using the strength of the alliance in Britain and indeed our members, the network of our existing members, and also to encourage the formation of similar alliances in other countries, which is something that this alliance will be very happy to support. We will continue to promote the scheme to the trade and the public. We will do everything we can to improve accessibility and to remove barriers, whether they be lack of technical knowledge, trying to find financial support so that even the smallest businesses are not excluded, and as I said, to develop those incentives and to look at what else is out there internationally. So we're working with the AIPH and, as I said, talking with other certification bodies. So just to summarize all of that, as everyone on this call knows, plant pests and diseases are a serious and growing threat. Tackling it will only be done effectively if we have a combined approach between government, the trade, and that means all sectors and especially growers, and the public. The principles of the standard provide a robust and proactive framework, not only to put that good practice in place, but to provide recognition. We have a very flexible approach to working with other schemes and bodies to help improve uptake and to remove the barriers. Again, repeating the mantra there that we want everybody to understand the risks and the threats and what can be done to mitigate them. We want as many people as possible to apply those principles, and where applicable, for people to demonstrate through certification, and then to use our strap line together, we can be plant healthy. So finally, by way of follow-up, do have a look at our recently relaunched and constantly evolving website. We're trying to make that a hub as far as possible for resources to do with plant health, and we're always happy to hear with others with material to share. That's the URL. Don't just take my word for it. There are a number of videos on the site in which you can hear existing members talk about the scheme. If you're interested in following up on any aspect of that, that's my email address. I'm director@planthealthy I should just say the website is currently only in English, but translation is very much on the pad. And above all, spread the word. We talked about awareness being the most important thing, and awareness of the problem and what you can do about it. So it's great. There are 400 people on this call. I think there are more who have signed up who presumably will get the material. And if you all talk about it and spread the word, then that's probably the most powerful thing that we can do. And at that point, you'll be delighted to know that is the end of my presentation. Thank you very much.
Thank you very much, Malcolm. Great. Before we're going to start the part where we are answering the questions and comments that you put in your chat, I would like to thank each and every presenter for the great talks and the information that they've given. And I think, you know, this was a really good start, and it gives a great overview, and it also inspires, you know, to continue our work towards healthier nurseries. Okay, now if you have questions that are coming up during the discussion, please continue to put that into the chat. I would like to start with the first question, and it's a bit general. It doesn't really say who it is for, but it asks about possible sustainable approaches to manage Phytophthora under field conditions, so away from the nurseries. Any suggestion would be greatly appreciated. Now, I think, you know, maybe Tyler or Sarah could volunteer to address questions beyond the nursery.
I think maybe I take the lead here.
All right, okay. You can start, yes.
I don't think you can see me now, but I think it's not.
We can hear you.
Okay, now you can see me. The management of Phytophthora in forest stands in the field is a complicated matter. It's a particularly complicated matter here in Europe because our hands as pathologists are bound now, and we have hardly any chemicals anymore to fight fungal diseases or oomycete Phytophthora diseases in the field. In other continents, phosphite is used, potassium phosphite, on a large scale, which is sprayed from helicopters or planes in Australia over eucalypt forests, and they have halted and slowed down the spread of Phytophthora cinnamomi, the bulldozer through the eucalypt forests in Australia. This is a very efficient method. We also proved in many trials, and Bruno has also done some of these trials here, that it is a very efficient method also against Phytophthora in forests in Europe, but it is no longer allowed as a fertilizer. That is a problem of the European Plant Protection Law. Our hands are bound there. We can do some scientific trials, but we cannot really apply it in the field anymore. So prevention of introduction is the most important thing. Prevent soil compaction of any way. Don't go with heavy machinery into the stands. Don't leave the pathways with the heavy machinery, because soil compaction leads to waterlogging in the soil, and waterlogging leads to spread of soil spores. Then there's something you can do with the next generation of the forest you plant after this harvest of this forest, and you should use there tree species in the next generation that are more tolerant to the Phytophthora that you have already on this site growing, etc. So these are things you can do, but you cannot do a lot in Europe. But I give the word also to Bruno, because Bruno has done a lot of this management research in Sardinia. Bruno.
No, Thomas, I can just confirm what you said. I think the best way, the best approach to manage Phytophthora in the field is to prevent its introduction, because once Phytophthora is introduced in a new site, it's very difficult to manage. There are different experiences, as Thomas said, but it depends on the context, on the forest, on the forest management, on the susceptibility of the host. Within a live project which is called Phagesos, we have developed an integrated protocol that include some treatments with biological controls like Trichoderma strains, Bacillus as well, and also some biostimulants, which are extracts from plants that act as a biofumigants, and they are allowed to be used into the forest. But we also, in these integrated protocols, we include some measures to prevent also the spread of a Phytophthora within the sites. And I think it was Thomas was saying about selecting tolerant genotypes maybe will be in the long run the best way to restore areas treated by Phytophthora, because if you replant the same host and it is susceptible to Phytophthora, then you are not really restoring. So probably selecting tolerant genotypes to Phytophthora, and there are a couple of papers in the last years would be very useful to manage Phytophthora into the forest.
Okay, thank you.
And I don't know if I can add something related to agriculture, because in agriculture maybe a bit different, because we have tolerant rootstocks to Phytophthora in some of the crops. Some of the main crops you can, through plant breeding over the years, they have produced tolerant rootstock, and that is the only way sometimes we can grow these crops in soils that they had Phytophthora, but they can thrive. I have to say as well that lately we have extreme weather events where we had a lot of flooding and the trees were underwater, for example. We had a couple of years ago, and in these situations, even that the rootstock was tolerant, the variety it was not tolerant, but the water level was above, to the branches level, and then some infection occurred. In the case of agriculture, I think there are more work maybe on plant breeding for tolerance to Phytophthora.
Yeah, just to quickly add, I don't want to linger too long on this question, but it's a great question. I mean, you could say a lot of maybe most California agriculture was just finding tolerant rootstock to grow Mediterranean plants with irrigation. That's the success of California agriculture in many ways. But I think then there are success stories, arguably, in California and Oregon with Port Orford cedar and Chemosiporus lawsonii, which you've heard about, and Phytophthora lateralis. But I would say, I would agree with what Thomas and Bruno said, which is that prevention is so much better than the amount of energy and resources it takes to create one Phytophthora resistant plant, and the disturbance that happens when you have to go out and plant them is really devastating. If we could prevent that, it's so much better in the long run.
Thank you. Sarah, did you want to add anything to that?
Yeah, no, I suppose I misinterpreted the question a little bit, because I thought they were talking about nursery field production, but obviously it's around woodland. Presumably it's about woodland. And all I can say is, in our experiences that we work with sort of in commercial forests with Phytophthora morum, it's really about phytosanitary felling of infected forests to reduce the spread. But our experiences with juniper is actually very interesting. Juniper is a native species being killed by Phytophthora ostracedrae, and we are finding natural levels of resistance and tolerance in native populations. And so now we're advocating, working with site managers, not to plant, because that is a high risk of introducing new genotypes of Phytophthora ostracedrae, but instead to manage the sites for natural regeneration. These are obviously existing juniper sites. We're not talking about completely new woodland creation here. It's about, preventing supplementary planting of already existing populations and relying on the fact that individuals in that population will have natural resistance and allowing them to propagate themselves. So we're doing a lot of work now on the genetics of resistance and understanding how we can work with that to facilitate population recovery in a native host.
Okay.
Andrea, maybe we can jump a little bit around in the questions, because there was another question that relates a little bit to this, what to do in the field. There
Yes.
was a question by Frank Schumacher from the Botanic Garden, University of Vienna, and he was asking, how is the situation estimated in dry area grasslands and shrublands? It seems water and floods play an important role, but example of Sardinia area with juniper or olea in Spain relates also to dry situation. Is that correct? And we saw the same also applies to the heathlands in California. To say brief, in the past, Phytophthora were called water molds, and everyone thought this is just related to very wet sites. But what we know now, after we have studied in detail 270 Phytophthora species, and we have much more species undescribed and studied, we know that Phytophthora is an incredibly variable genus, and we have adaptations to any kind of ecological situation. And we have Phytophthora species that are very well adapted to extremely dry, semi-arid climates even. So the situation with junipers and olea in Sardinia shows that some Phytophthora species, they can survive droughts very well in their resting spores in the soil. And if there is a heavy rain coming or a long-during rain coming, then they explode in inoculum, they kill the root systems, and afterwards these damaged root systems are not available for the tree to take up water during the following droughts. And this interaction between droughts and the Phytophthora damages is actually what is really lethal. And we once described the Phytophthora species from Western Australia. We called it Phytophthora arenaria, from sand. Yeah, it was a sand Phytophthora because it was in extremely arid heathlands naturally where it evolved. And it is laying in its spores in the soil, in dry, very hot soil for 10 years or longer. And then every 10 years or 15 years, a tropical cyclone comes down the Western Australian coast and floods the whole area within 1 week completely. And that's where the Phytophthora wakes up after 10, 15 years in the ground. It produces masses of sporangia at the same time that the roots of the trees are multiplying because they have to grow when it's wet. And then they're all killed by this Phytophthora. And then it's dry again, and the Phytophthora is again for 15 years in its resting spores. So we have perfectly adapted Phytophthora species to very dry, arid climates.
Thank you, Thomas. I think we might just, there's another question related to sort of field situations, because Unite asked basically Thomas and Bruno Scanu about if there are Phytophthora species occupying forest natural systems and then transform to agro-ecosystem as plant destroyers. So basically, what is the risk that those Phytophthora that are out in the natural ecosystems then change host range to become then the plant destroyers in agriculture, if I understand the question correctly.
I'm sure that other speakers can add something to that, but I want to add, because this question came from someone in Indonesia. We were working in Indonesia and we could see that there were the rainforests which harbor a huge diversity of Phytophthora species when they are cut down and replaced by plantations. And that was in the past, for instance, cinnamon plantations. And then the Phytophthora jump onto the crop species that are not adapted to these Phytophthora that are growing in these native soils, and they are killed. And that's actually where Phytophthora cinnamomai was first described in 1922. And 100 years later, we went to the same area in Sumatra, and we were again in this area looking at the rainforests, which were loaded with Phytophthora cinnamomai. And in the middle of this rainforest were the cinnamon plantations full of bleeding cankers on the bark, and they had exactly the same genotype of Phytophthora cinnamomai. And a similar situation is with the eucalypt plantations, which are also planted on millions of hectares of previous rainforest area. They are killed by Phytophthora cinnamomai, and the disease is called little leaf disease. It's exactly known as a Phytophthora disease. Maybe someone else wants to add something. Bruno, you have lots of experience with the interaction between agroforestry systems and agricultural systems with the olive trees, the native olive trees, and the planted olive trees.
Yeah, I can tell you about the wild olive forest that we have in Sardinia, and we recently found a severe outbreak of disease caused by Phytophthora. But in this case, actually, the native forest, the natural forest where Phytophthora was actually introduced using a cultivated olive that were planted within this wild olive forest. So the way of introducing Phytophthora was from agricultural plants into natural forest. Yeah, that's the only example that I know, but it can be also that some native Phytophthora species maybe jump into agricultural crops, as Thomas was saying.
Okay, thank you. And there's another question here from Tulin Ertek from the Plant Protection Central Research Institute. And the question was, why do some trees in the same area appear completely dead due to the disease and some others remain healthy? Such as, affected by Phytophthora ramorum in oak forests in Sardinia, or in Patagonia by Phytophthora ostracedri. So there seems to be a big,
Just I can quickly, I want to shout out that restoration sampling guide that I highlighted, because this is a really interesting question, and there's a lot of factors, right? Trees. A big mature tree in a forest can lose more than 50% of their roots before you see above ground symptoms. And in fact, Thomas really alluded to this issue as well, or one reason this could happen where you have repeated floodings and then roots are rotted during the flooding, and then when the water comes back, there's no roots left, and that's when the mortality happens. But it's a very difficult to sample for soil-borne Phytophthora because the above-ground symptoms are delayed quite a lot based on compared to the actual infestation. But that's part of the fun of Phytophthora diagnostics.
Okay, Sarah, you have.
Can I just, so when I saw that comment, the first thing that jumped into my head is that when you're looking at an invasive, our experience with juniper and ostracedri are that if it's a native population of host, you should have some resistance. If it's a genetically diverse native host, there should be resistance in the population. So my understanding would be that there will be probably resistant Ostracedrus chilensis trees in Patagonia, for example, that do have natural resistance to Phytophthora ostracedri, and that's probably the most likely explanation for them surviving if they are in a stand where there's heavy mortality around. So they haven't just escaped the infection, that they've been exposed to it and they're not succumbing.
Okay, interesting. Another question comes from Elham Padal-Sadiha. It's a PhD student from the Linnaeus University. And the question is this BMPs financially actually viable for nurseries? That question is for Bruno, but I guess maybe also Malcolm or maybe Sarah, or, what is your take on that? Financial viability.
Yes, if I can, this is probably the major critical points of best management practices. I think this has been highlighted by myself and also Tyler. It is especially the transition between conventional nursery and those using this best management practice is quite costly, especially when we use a steam for soil sterilization. These generators are very, very expensive, and also buying those benches to grow the plants are also quite expensive. So in our case, in our project we were working with public agencies which can afford this big investment. But if you work with private, maybe it could be more complicated, or at least this transition can be longer than other situation.
Malcolm, do you want to add to that?
Yeah, I guess what we would say this, wouldn't we, is that you have to think about what is the cost of not doing it. Just starting to look at the principles involved in terms of assessing the risks and the measures you can take in itself is not an expensive process. That's mainly around time. Certification can cost some money because there is the cost of an independent auditor coming in, but we provide support with that in terms of preparation. There are mechanisms in place whereby a group of growers can come together for their audit, so that's another way of offsetting the cost. But making a start is the most important thing. that systematic review. I get that some of the practices that might then need to be adopted, things like steam sterilisation, there may be some costs around that, but don't let that put you off making a start.
But is there any discussion about having compensation schemes, you know, supported by the governments? Yeah.
Yeah, so I mean, there are two sort of routes for that. One is for individual governments to look at what support they may be able to provide, and that does exist in some countries, not many. What we're looking at is a private scheme, which would be a commercial insurance for people who had completed their certification and who had a current valid certification against the plant health management standard that would provide some support. And that's not certain at this stage how long it will take, how we might be able to roll that out in different countries. A lot of factors to discuss, but our early conversations suggest that that is a possibility. There is an appetite to offer that kind of support.
Okay. There's another question regarding immunity or susceptibility of plants by Alexandra Jankovic from the plant protection, as she's a plant protection engineer at the public utility company. That I think goes to Bruno, but also to our other speakers. Does the occurrence of the disease caused by this pathogen depend on the plant's immunity and the variation in resistance?
I would just like to say for Phytophthora, it's the least amount true for Phytophthora. In general, if you ask phytophthorologists, they will generally say almost every plant is susceptible to almost every Phytophthora under the right conditions. And so while there are a few conditions, Sarah mentioned them, it actually really is related to the Cupressaceae largely. In other conditions, for example, people have been looking throughout California and Oregon for natural resistance in tan oak, the major susceptible host to sudden oak death. They have not found anything, right? And there's a huge natural experiment ongoing, right, where millions of trees are dying. And to the extent anything has been found, it's what's called quantitative resistance, qualitative resistance. It's a resistance that's very difficult to breed, even if it is durable. So I would say that Phytophthora and resistance, it's not the right way to think about it. Generally, if I was thinking about a risk analysis, you have to imagine that every species of plant could be susceptible to every species of Phytophthora. And in fact, one more thing, that's actually really validated by the molecular basis of plant immunity. If you understand resistance genes, effectors, these kind of things, that completely explains why Phytophthora is the plant destroyer.
I mean, we have to differentiate between co-evolved Phytophthora and hosts and non-co-evolved. Most of the Phytophthora species we are talking about today were introduced invasive non-co-evolved, not co-evolved with the hosts. These are naive hosts. They know nothing about these Phytophthora, and that's the problem why they are so susceptible and have almost no resistance against Phytophthora. If you take the few native Phytophthora species you have in each region, they are co-evolved with the Phytophthora there, and we saw this with our expeditions to Asia, in the rainforests in Asia. There you walk on Phytophthora cinnamomi in the rainforest. You walk, in every step you walk on Phytophthora cinnamomi, and you don't see any symptoms in the rainforests. And then you have the cinnamon plantation 5 meters beside the rainforest, and the trees are dying there, because they have never seen a Phytophthora cinnamoma, and they have no tricks to counteract the tricks of Phytophthora. So the problem is the co-evolution. And what we can say about our North American, Australian, New Zealand, European landscapes where we introduced hundreds of Phytophthora species, most Phytophthora species will kill most of the plants there. Of course, there are differences in the forest. There are some genotypes that are a little bit more susceptible, but there are also trees simply, they are lucky escapes. Yeah, they were simply by chance not hit by inoculum by that time now, but they will die in a few years later. There are also differences in microsite conditions. In every forest, you have different soil types with more waterlogging, less waterlogging, a little bit of ponding in a depression, and this tree will die because Phytophthora produces lots of zoospores. So there's many reasons, but in the end, very often trees also die because Phytophthora opens the gates and secondary fungi, secondary parasites come in and kill the trees. So it's also depending on which secondary parasites are actually in the rhizosphere or in the vicinity of a certain tree. And some are simply lucky because there is none that takes over and kills them.
I think, you know, that's a very clear answer on that. I was just wondering, we can stay a little bit on the management side. We have some questions regarding biological strains. You know, if there's some microorganisms, the microbiome of the plants, is that what we have destroyed? Is that basically why the invasive species are so destructive? You know, I like to hear, you know, several opinions on that.
I mean, this is a big market now. It's a big market to sell mycorrhizal strains. It's a big market to sell trichodermas, Bacillus subtilis, etc. So this is a market for the industry. So that they promote these products as being the solution against Phytophthora or any soil disease is clear. It's in their interest. But what we can also see is in most sites where you actually do an analysis where the mycorrhiza is actually really missing, it is not missing. The mycorrhiza is still there in the forest on the fine woods. You just have to look at them, and they are still dying because mycorrhiza is not necessarily protecting against Phytophthora. And there was a lot of research in Australia done in the 1970s with Phytophthora cinnamomi, and they stopped this in the 1980s because everything that worked very well in the lab, when you applied it in the field, the situation was much more complicated and it didn't work very well. Now it's about the microbiomes, and I'm sure that we damage microbiomes with all the influences we are doing, climate change, pollutants, etc. But also microbiomes will not be the solution. It will be helpful somehow, but it will not be the solution for the forests. What we can say is that they are supportive, and Bruno will certainly agree to that. If you put trichoderma strains or bacillus strains in nurseries, they will help these plants to get stronger and cope a little bit with Phytophthora. In these 2 years, the plants are growing in the nursery. But it is not the solution on the large scale for hundreds of millions of hectares in North America, Australia, or Europe.
And I'll just quickly add, you know, forest pathology is very complicated, and there are a lot of emerging diseases that are caused by a variety of factors, and they maybe interact with climate change. Generally, if you see Phytophthora and you see sick plants, that's generally the cause. And it's generally because humans move them there. And there are plenty of exceptions, or at least a few, but it actually makes your job as a forest pathologist a lot easier if you study Phytophthora, because generally it is the cause of the problem.
Okay. Any other opinions on that? Yes?
If I can add, just to confirm what Thomas was saying about the biological control, I think it's there is a lot of effort from the scientific community because of now we cannot use any chemical products, especially in Europe, in forest, and even in agricultural crops. As Thomas said, actually, there's some biological control like trichoderma or bacillus, they work very well in vitro conditions and also in mesocosm triads, where we have a simplified system with the plants, the pathogen, and the biological control. But then when we move into the forest, especially, which is a more complex situation, it's difficult to assess whether they are efficient or not in suppressing the disease. But definitely in nursery, I would suggest to use some biological control, especially in the first phase of growing seedlings. They will definitely help the plants, and particularly if we have a sterilized potting media, they can develop and protect the roots in some way.
Yeah. Alexandra Jankovic from Serbia is also wondering, maybe good advice would be to encourage production in one's own countries and to control it well and to promote the planting of those domestic species produced locally. Is that basically some part of the take-home message?
I mean, I think this is one of the main messages here, because the globalism in plant trade internationally, overseas, but also within Europe, has become a problem. It's clear that we are exchanging pathogens. They arrive in one port of entry and then they are multiplied in a very very, very quick time all over Europe, and the same certainly applies also to North America, because of the plant trade. So producing locally, especially forest nurseries, is the best. That's clear. You keep out a lot, and pathogens can only move very slowly if you don't have international trade. Clear.
Okay. No more comments on that. A few questions here about, maybe seed treatments. If how much does it help to do seed treatment with hypochlorite, for example, disinfecting seeds before you plant them, and then plant them in healthy soil, I guess, does that have an effect?
Even better is collecting the seeds. If you can collect the seeds before they've dropped on the ground, because Phytophthora is generally not aerial, those seeds can be considered to be clean. So that's a possibility. And we've done some studies in California, not published, but that essentially if you collect anything above a meter off the ground, you're not going to get any of the Phytophthora that's in the soil that would be on there.
Yep. In terms of, there's also the question, does Phytophthora actually survive in salt water?
That's an interesting one, because we had a project in Europe the last 3 years. Restore Seas has also a very good website. It's just finished. We made surveys in marine ecosystems all around Europe, as we did also in Asia. And we found more than 140 oomycete species. Of these, half of them Phytophthora species in the sea. So Phytophthora species can survive very well in the sea, sporulate very well in the sea. Some species are even marine. You only find them in the sea, like Phytophthora chesapeacensis or so. And they infect also seeds of Zostera plants, root systems of Zostera plants, seeds of seagrasses, leaves of seagrasses. And in the seeds, there are a huge problem, which was shown by Dutch researchers, where 90% of the seeds of Zostera in the seas outside of Holland, offshore, were infected with Phytophthora. That's a problem. And they could not be eliminated with surface sterilization with copper fungicides, because the copper does not move into the seeds. So yes, Phytophthora survive very well. We also found once Phytophthora cinnamomi from a floating leaf in the Indian Ocean. So they survive very well.
Okay. Thank you. I think we are almost coming in for landing. There's one comment from Thomas Brandt from Lower Saxony, Plant Protection Service, Germany, who's commenting on that. Yes, we have, we're focusing on preventive measures, but once it is established out there in the field, there's not much we can do. Is there something you can, you can say to reduce the feeling of helplessness when we're going out there and say, yes, this whole area has been contaminated. What should we do?
I think we already answered that in a long discussion in the beginning of this discussion section, that there's not a lot you can do actually in forest and natural ecosystems. And I think I just referred to what we said before. Yeah.
Okay.